So I'm going to get started. Most of you will know I'm Daniel Heath. I'm one of the primary PIs on GEN-FISH, Genome Canada Network, technically an LSARP grant. Steve Cook and Margaret Docker are the other two primary PIs, and there's a total of 23 PIs involved in the GEN-FISH mandate. Before I start with my welcome remarks, I want to give a land acknowledgement. GEN-FISH acknowledges that our universities, partners, and our research benefit from, and in fact are situated on the traditional and unceded territories and lands of Indigenous nations. We acknowledge the rights of First Nations, Métis, and Inuit people of Turtle Island, and I encourage all of you to reflect on where you are situated in this broad national network and the history of where you're situated, recognizing that participants, all of us, are on different territories with different histories and different traditions. Good. I'm going to keep my welcome comments short, but I will ask you all to cast your mind back 4 years ago, just about this time of year. We had our first GEN-FISH meeting where we were all here in Windsor in person. And I don't know how much you remember, but I look back at my notes. We had a very ambitious plan. And even then we were, some of us more than others, but concerned about the possibilities of actually completing. We are concerned about the fact that the GEN-FISH action plan involved development and applications are critically dependent upon the development stages. But we still thought we could do it, and we were confident we'd make a big difference for Canada's freshwater fishes. And then, of course, COVID hit. And we basically were locked out of our labs. Our HQP were provided with make work projects in general. Development slowed down. Field work virtually halted for over a year, running up to 2 years. We're now at 4 years. We've been given an 18-month extension. Normally, we'd be shutting down GEN-FISH as of January 1st, 2024, but we have 18 months no-cost extension. It is remarkable what we've accomplished in terms of people trained, our HQP, in terms of partnerships that we've developed, and in terms of the amazing genomics technology that we've developed, and not just developed it to be used in laboratories at research institutes and universities, but developed it in a framework that's going to make it useful and ultimately applicable for all of our end users, partners, and potential stakeholders. We still have work to do in our 18 months. I suspect the ramifications of GEN-FISH will continue on into years past, and potentially even Genomics Canada and provincial support extension of the funding. But now what I'd like to do is launch the GEN-FISH AGM for 2023. We're going to start with HQP presentations, then we'll have a break, and then we're going to move into roundtable discussions about some of the key components of what GEN-FISH is all about. But I will remind you that GEN-FISH AGM 2024 will be our last AGM, and we're planning to get together with all of the PIs and the HQP to come up with a plan where we can have an in-person, hopefully, AGM that will really tie up GEN-FISH in a way that will be exciting and important. So I'm now going to pass this over to Paige, who's going to introduce the HQP speakers, and we'll go from there. Thanks very much.
Thanks, Daniel. Yeah, our students in HQP submitted some abstracts in a competition for one of these coveted 15-minute presentation slots. So first up on deck, we have Peter Martin.
Hello, should I share my screen now?
Yes.
Okay. All right, is that looking good? Hello, everybody. I'm Peter Martin. I'm working at the University of Guelph, and today I'm going to be talking to you about the development of a decision-making model for evaluating methods of sampling fish in inland water bodies. So, what is this?
Hi, Peter, sorry. We
yes.
just see your, we're not seeing the presentation view of your slides.
Oh, let me see if I can fix that. How's that? Is this sharing now?
Not yet.
Okay, apologies for this. How about now?
We can see your slide outline and notes.
Let me see what I can do. And this is this working now or not?
I see a black screen.
Okay. Well, I may just present from this view then, if that's all right, since it doesn't seem to be working otherwise. So to reintroduce myself, I'm Peter Martin from the University of Guelph, and I'm going to be talking about the development of a decision-making model for evaluating methods of sampling fish in inland water bodies.
We're not any slides now, Peter.
Oh, so we're seeing nothing now.
Yeah.
Do I have permission to share my screen? Because I should be sharing it.
Yep. Yep, permissions are all fine.
Okay.
Because you were sharing the full outline before.
Is this currently sharing the outline?
No, just sharing a black screen.
Okay, apologies for this. So currently it is not sharing.
Right.
Correct.
Okay. This has always worked for me in the past.
Peter, you're also welcome to send them my way and I can maybe share.
Yes, I can do so if you'll give me a minute.
Okay, who's got a good fish joke to keep us busy for a few minutes? Take the pressure off of Peter. Usually I'd say Dennis could do it, but Dennis is not here right now. He's usually got a good joke up his sleeve, although it's not always appropriate.
So Oliver, since nobody's volunteering a fish joke, how about a bird joke?
Why did the researcher decide to study birds instead of fish? Because we all know birds are easier, get way more money, get more easily published. There you go. It's not really a joke. It's just an unfortunate fact. There you go.
Okay, I've got some fish puns. Are you ready, everybody? Why are fish so easy to weigh? Because they have their own scales.
Yeah, I was going to say, I'm sure it has something to do with scales. Yeah.
What do you call a fish who practices medicine? A sturgeon. Come on, people, help me out.
Andrew in the chat has one. A fish walks into a bar. Bartender says, "Whoa, how'd you do that?"
That's a good
Okay, that
one.
one wins the prize so far, I have to say.
I agree.
Yeah.
Declan's got one. This is not the time nor the place. You need to know the spelling of it. So that's awesome. Thanks, Declan. Ken.
Okay, ready? Two parrots are standing on a perch. One parrot goes to the other one. Do you smell fish?
Oh. That's really good.
I don't even think I have any lamprey jokes.
I don't know, Margaret. I think the classic was when Joe asked you about what species of lamprey was in the Star Wars movie.
Yes, indeed. Yes.
And Joe Ackerman had come in late to Margaret's talk, so Margaret said, "I already covered that."
Yes, that he was, yes, because he was asking about, I guess the opening of the Empire Strikes Back, which I had not seen. It was for a job. It was a job seminar, so I was a little bit nervous to begin with. And he starts with, maybe I missed this, but yeah, what species of lamprey was, in the opening of the Empire Strikes Back. And because I didn't have any other answer, I just said, well, yes, actually, I did cover that already.
But there's only 3 of us in the meeting here, Daniel, that actually remember that. But I must say that was a stellar performance, Margaret.
Yeah, that was a while ago, wasn't it? It's funny how time flies.
Brooklyn, I say it with love in my heart. So don't worry.
It's a risky one, Tina. That's pretty risky.
Uh-huh.
So I have sent the slides to Paige.
I should have had a Kahoot quiz to fill the time.
Oh yeah.
Or better yet, an economic game where we all win money.
Yes, yeah.
So Paige, do you have Peter's file yet?
Nothing yet. I'm refreshing like mad.
Peter, how big is your file?
It should not be too large.
Okay.
I really apologize for this. I've not had this issue in the past.
Peter, what screen were you sharing when we could see the whole file layout?
I was sharing the,
Because that one we could see. We did. I mean, it just wasn't a
yes.
presentation layout,
So
but we could
so
see.
if I'm sharing like this,
Yeah,
are
we
we
see.
able to see it?
No, we see a black screen.
Okay, because that was how I began sharing. If I do this, is there any change?
Yes.
I have mine up.
There you
sorry.
go.
Oh,
Awesome.
Oh, that's Paige. Wait, who's, yeah.
I'm sharing at this point. Sorry, that was
okay,
bad
cool.
timing.
Okay, good.
Okay. Let me see. Okay, and I'll just make a note when to change slides. Okay, so let's begin. This is a talk on the development of a decision-making model for evaluating methods of sampling fish in inland water bodies. So, next slide. What is this? This decision-making toolkit seeks to accomplish several simple goals. When a wildlife survey is designed, the choice of sampling method used is an important one. For sampling fish populations, there are many different sampling methods, each with their distinct advantages and disadvantages. These advantages may be in terms of how they sample, being better in certain environments or better at identifying certain species, or logistical advantages, having lower costs or being faster to implement. With all of these considerations, it can often be difficult to make an informed decision on which method or methods to use. This toolkit aims to provide a comparison between sampling methods given the specific logistical and sampling requirements for a project. It aims to provide a consistent output requiring only simple user input. And finally, it is intended as a rapid first pass for decision making, serving to highlight the most viable method or methods, as well as those unlikely to be suitable. Next slide. Before we begin, certain terms relating to decision making should be defined. A choice is an option with a collection of criteria. Here, the sampling method, electrofishing, is a choice. Its criteria are cost, efficacy, perhaps rated out of 100, and timeliness for completion. These criteria are what are used to evaluate and rank choices. A preference is a statement such as cost is much more important than efficacy. These preferences are expressed by users and, as one expects, lend greater or lesser importance to a given criteria. As an aside, the numerical values in this talk are just for demonstrative purposes and are not necessarily 100% accurate. Next slide. So, why is a toolkit necessary? We can imagine a simple choice between two sampling methods, say dip netting and electrofishing. If the only important criteria is cost, this decision is very easy, with the less costly choice being more desirable. Next slide. If there are two important criteria, here cost and efficacy, the choice may be more difficult if each method excels in one way or another. This is where preferences come in. If a cost is more important than efficacy, then the decision is once again simple, with dip netting, the less costly method, being preferred. Next slide. But that isn't the end of the story, even in the two criteria, two choice case. It may be that there is a small difference in one criteria and a large difference in another. Here, dip netting is only slightly advantageous in the more important criteria of cost, whereas electrofishing is much more effective. It's natural to assume at some point a great advantage in a less preferred criteria will outweigh a slight advantage in a preferred criteria. This tipping point depends on the strength of a preference. Is cost much more important or only slightly more important? Next slide. This becomes even more complicated when we consider adding more choices and more criteria to consider. In this example of 3 choices and 3 criteria, depending on the preferences relating the various criteria, any of the choices could be found to be ideal. Is it important to excel in a single most important criteria, such as eDNA sampling excelling in efficacy? To be most favorable across a number of criteria, such as dip netting being the least costly and least time-consuming method? Or is it best to find a middle ground, such as electrofishing, which lies in the middle of all 3 criteria? The decision-making toolkit will allow for these complicated decision-making models to be easily developed from simple questions on user preferences, as well as statements about the project itself. For example, what sort of water body is being sampled, how many sites will be visited, etc. So let's move towards a better way to make decisions. Here we're using an MCDA model, which stands for Multiple Criteria Decision Analysis. And this framework provides us with several proven models for decision-making across a wide variety of circumstances. At their core, these models are linear algebra frameworks which convert a set of choices and preferences into a ranked list, as we have been discussing so far. The method being used in this toolkit is a form of analytic hierarchy process. Without diving too deeply into the math, at the top level of the hierarchy is our goal. And I forgot to say next slide. There we go. At the top level of the hierarchy is our goal, selecting a sampling method. Next, there are the criteria being used to make this decision. And finally, at the bottom, there are the choices of sampling method, which are evaluated based upon each criteria. Next slide. The bottom level works as the simplest decision model we saw earlier, where only a single criteria is considered at a time. Let's highlight the cost of each of our options. For each criteria, the choices are given a score between 0 and 1, which is weighted based on the scores of the other choices for the same criteria. Here we see the scores for costs of $5,000 and $7,500. Note that these scores are calculated without considering any other possible options. It's also important to see that these scores are dependent only upon the proportion of the costs for each choice. Next slide. Thus, we see that multiplying both costs by the same constant, here too, has no effect on the scores. Next slide. Whereas changing only one of the costs, lowering the cost of netting, will have an effect on both scores. Next slide. In all of these cases, the scores will also be scaled based upon the preference given to a criteria. Here, preference is given to cost. That is, cost is considered to be more important than efficacy. Next slide. However, if the reverse is true, the scores will change, becoming lower as cost is deemed less important than efficacy. It should also be mentioned that the degree to which the scores are scaled depends on the strength of a preference, with stronger preferences causing more scaling to occur. Next slide. With level 1 finished, we move up to level 2, where we begin to consider multiple criteria. The best option for each criteria is identified. Because a low cost is desired, that is, cost is being minimized, the lower score is chosen. With efficacy, we want the most effective sampling possible, so the score is maximized. In this example, the best score in both cost and efficacy belong to netting. Next slide. But this domination by a single choice is not generally the case. It is more likely that a variety of choices will be represented in the best scores. In addition to the best scores, the worst scores for each criteria are also identified. When there are more than 2 choices, most options will naturally fall somewhere in the middle. Next slide. The final step in this process is somewhat involved, but at its core, a measure of distance is defined. Each choice is evaluated against the theoretical set of best possible scores, as well as the theoretical worst scores. In each case, the distance represents how close to ideal a choice is, or how close to the worst possible option it is. A choice which dominates across all criteria will have a distance from the best option of 0. Next slide. A final score, again between 0 and 1, is assigned to a choice as a whole based upon both measures of distance. Using only a single measure, for example, only the distance from the best option, is not enough to ensure consistency in all cases. Next slide. These final scores can be used to assign a preference ranking to the choices, with the highest score being the most desirable choice. A few points should be made about these scores. Firstly, they are relative. A certain set of choices and preferences may have 0.6, for example, as the highest final score, where another may have a highest final score of 0.9. Values should not be compared externally. Again, these are relative measures of desirability within a certain project. A choice being well suited for a single circumstance does not speak to its suitability in other cases. Secondly, these scores can provide useful information beyond simply ranking options. Two final scores which are very close together mean that the rankings are sensitive to slight changes in preference of criteria or in the criteria themselves. Small changes in cost or efficacy, for example, may alter the rankings in favor of one choice or another. While these cases are one of the reasons this model should not be used as a final step in decision-making, the ability to highlight these situations is one of its greatest strengths. Next slide. To illustrate this point, we can examine the dynamics of these final scores. How they change is one of the criteria is altered. Here we see how the final scores, measures of desirability, change as the number of sites being sampled changes. Number of sites sampled does not change the efficacy of sampling, but it does affect cost, and it affects it differently for each sampling method. It is clear that between, say, 13 and 17 sites are being sampled, careful consideration must be paid to the choice of sampling method, as there are two strongly viable options. This feature is difficult to reveal in the absence of an MCDA model. Next slide. We can also see how these dynamics change. Here, the only difference compared to the previous graph is that a preference is given to the criteria of cost rather than that of efficacy. In this case, a single method is preferred across a wide range of project scopes. Next slide. With the decision-making model developed, it must also be presented in a user-friendly toolkit. Users will supply information about the sampling project being examined, and all of the math is performed automatically. For the most part, the type of user input required is simple and can be classified into three categories. The first is that of logistical considerations. These are variables which are expected to have an effect on the cost of a given method, but not its efficacy. For example, the hourly wage of sampling technicians will increase costs, as will the number of sites sampled within a project. The second category is site considerations. These are characteristics of the water body and wildlife being studied. These are expected to have an effect on the efficacy of sampling methods. While logistical considerations are never expected to affect how well a sampling method performs, site considerations may have an effect on cost. Site considerations may include the type of water body, whether lakes, streams, or wetlands are being sampled, or how well studied a site already is. Currently, the list of site considerations is limited. However, this list aims to be expanded to include the species of wildlife which are being studied, as well as project objectives. Unlike logistical considerations, which are numerical, these site considerations are categorical. The final type of user input is preferences. In this early prototype, only cost and efficacy are included. Typically, these preferences are expressed as a pairwise comparison between criteria. For example, here, efficacy is slightly more important than cost. Comparisons must be made between each pair of criteria. For example, adding the third criteria of timeliness will require two additional comparisons between timeliness and cost and timeliness and efficacy. Next slide. The toolkit also seeks to automatically estimate cost and efficacy of each possible sampling method. Again, this is not intended to replace a traditional cost evaluation, but rather provide a ballpark estimate for decision making. The model used to estimate costs is based upon a statistical analysis of fish population sampling in Ontario. Fixed costs, such as the cost of sampling gear or lab costs, are combined with variable costs dependent on how the sampling is performed. For example, it may be more time-consuming to sample lakes compared to rivers, and thus the cost will be higher. Efficacy is estimated based on qualitative judgments obtained from a literature review. Due to the nature of these MCDA models, subjective judgments are often sufficient. Thus, we do not need to know how many more fish electrofishing is able to sample in a lake when compared to netting methods, only that one method is well suited to sampling in lakes and another method is poorly suited. Building a database of these subjective judgments is ongoing work. Next slide. This concludes a brief introduction on the development of a decision-making model. I hope I've highlighted why such a model is important, as well as how one can be developed. Thank you for your time, and thank you for bearing with all of those technical difficulties.
Paige, is there time for questions or should we go on to the next speaker?
If we want to have one question, I think that would be fine.
Well, I don't see any, so I'm going to ask a question. Oh, Tina, go ahead.
Oh, I don't want to take your question away, Daniel. Go for it.
Well, my question is straightforward. So determining those cost, efficacy, and timeliness metrics, do you see reaching out to practitioners, managers, fisheries biologists? Where do you think you get that information from, Peter?
I think ideally we want to collaborate with people who are out there on the field doing this sampling. And that's one of the big goals of this project, is that outreach piece and talking to managers, talking to people who are running these projects in order to get as accurate costs as possible, to get as accurate judgments based on how effective various sampling methods are in various situations.
And then, Tina?
I was just going to then take this opportunity, since we're all here together, and then if there's anyone here at our meeting at our AGM that would like to get involved with Peter and Dr. John Liebernois' work, Peter, can you please put your email address in the chat and we can get in touch with you? So it's just this is an open call for folks here at the AGM, or even to help Peter and Dr. Liebernois network with others. I think that would be amazing. We're also going to be talking more potentially about this decision-making toolkit tomorrow in our little workshops in the afternoon. So if you've got more questions, I think it would be a great place then to come as well.
Yep.
Thanks
I'll
a lot, Peter.
absolutely put my email in the chat. Yeah, thank you very much.
Thanks, Tina. That's important. The other thing is eventually modifying the model for the STP chip decision making as well. That might be a little more challenging, but in terms of generating the data. Okay, thank you, Peter. Paige, do you want to introduce the next speaker?
Yes, next up we have Julie Viana.
Okay, so just 1 minute. Oh. Okay, here. I don't know if you see something.
Yes.
Yes, okay. Yeah. Oh, I'm not sure. I don't know. Have you my notes if I yes, I don't know. I think that it's not a good screen. Okay. No, it's not good. It's better. Sorry. Okay, here. And now just, okay. No. Oh. It's okay. I'm not sure.
Julie, we don't see your presentation screen itself. We do see your PowerPoint.
I can switch my spot with you and then you can figure it out after if that's okay, because I think I'm after you, so if everyone agree.
Okay. Et si Julie, vous voulez envoyer ta présentation à Paige,
Vous voyez,
juste en cas.
Je l'ai déjà envoyé, mais genre, est-ce que là vous voyez le bon écran?
Non, on voit rien.
Ah, ok. Alors oui, effectivement. Okay.
So you would have to share it with us as you did with the previous participant.
Okay. So, I think that you have my presentation on email.
I do. I have it. Would you like me to share it?
Yeah. Yeah, I think that's better.
Okay.
Okay. So, hello everyone. Just to introduce myself, I'm a PhD student in my final years, and I'm carrying out a research project as a part of a France-Quebec co-direction. So my French supervisor is Fabrice Telechia for the University of Lorraine, and my Quebec supervisor is Céline Audet. So today I'm going to present some of the work I will be carrying out over the next few months on the use of transcriptional chips to study the response to anthropogenic stress in wild Brookshire population in Saint-Pierre and Miquelon. Next slide, please. So the Brookshire is a salmonid species and is one of the most popular fish for angling and aquaculture in North America. On Saint-Pierre and Miquelon, it's also the main freshwater species, and as can be seen in the picture to the left, there is a very close relationship between the archipelago fishermen and the Brookshire. But in recent decades, we can observe the general decline in wild populations due to habitat fragment degradation, overfishing, introduction of non-native competitors, or also parasitism. So this decline is set to worsen in coming years due to increasing anthropic pressures and also due to regional climate change, which is expected to overwhelm the adaptive response to the response capacity of this organism. Next slide. So I think that's yeah. I hope that my data will be directly useful for the project in which my thesis is integrated. This is the Ombles PM project that wants to enable the management of wild Brookshire populations. This project also aims to make a scientific recommendation on the best way to develop Brookshire aquaculture on the archipelago for restocking and also for human consumption, and also for the development of ecotourism. So next slide. When we talk about adaptive potential, we are referring to the ability of species or population to respond to changing environmental conditions by means of phenotypic or molecular changes. And I think that in Brookshire, the best known phenotypic diversity is probably that which leads to the expression of resident or anadromous phenotypes. And this phenotypic plasticity results in modification at different levels of the organism and involving morphological, physiological, behavioral traits. Next slide. Okay, but understanding the effects of human actions on the adaptive mechanism that's maintain wild populations is more complicated. And transcriptomic tools can be used to analyze the molecular and biochemical mechanism underlying the expression of functional responses to given environmental conditions. Next slide. And it's perfect because this is one of my research aims, which focuses on the identification of potential transcriptional fingerprints related to anthropogenic effect on Saint-Pierre-et-Miquelon, because this archipelago is made up of 3 main islands. Saint-Pierre, which is the most populated island with around 5,000 people and the most impacted by human activities. Miquelon, which has a population of 600 people and a moderate anthropogenic impact. And finally, Langlade, which is inhabited only in summer and has not seen any modification to its hydrosystems. So, next slide. So, to carry out this study, 10 hydrosystems distributed in several drainage on each island were sampled from May to August 2022 by electrofishing or traditional angling techniques. So on the island of Saint-Pierre, many hydrosystems have experienced a break in ecological continuity. And the 3 hydrosystems that were targeted were Savoyard. So sorry, I can't use my laser, but Savoyard, which has a dam in the upper portion of the hydrosystems, Tello, which has 2 former hydroelectric dams, and Capodiable, which has a waterfall just prior to the coastal area. In Miquelon, the Miron Pond in yellow, previously open to the ocean, was closed by a road constructions in the 1950s. But the 3 other hydrosystems under study were not human modified. And finally, Langlade have Brookshire populations that seems to have developed unique characteristics. The Ansaros hydrosystem is characterized by small Brookshire, Voile Blanche by extremely colorful individuals, and the Bon with the largest species captured every year. So we aim to capture a total of 30 individuals per hydrosystems, with 10 individuals captured in the upstream section, 10 in the medium portion, and finally 10 in the estuary portion. Next slide. A total of 230 Brookshire were collected on these 3 main islands, and following capture, fish were anesthetized with eugenol 100%, and their livers and gills were harvested and preserved in RNA later. Height and weight data were also collected for each individuals, along with a picture, as we can see to the right, with 4 individuals capturing 4 different hydrosystems. Next slide. Okay. Oh, sorry. And now I'm currently performing total RNA extraction on liver and gills samples. Once this first step is complete, I will retrotranscribe the messenger RNA into complementary DNA, and then use multi-species transcriptomic chips developed by the GEN-FISH consortium. And these chips are really useful because these chips include several genes involved in different physiological functions, as for example, pathway regulating growth and metabolism for liver samples, or also response to osmoregulation for gills. And once the data have been obtained, different statistical analyses will be carried out to explore the relationships between gene expression data and also anthropogenic effects. The next slide, please. Okay, so this is the end of my presentation. Thank you for your attention, and I will especially thank the fishermen of the archipelago, because when I arrived to take my sample, I really didn't know how to fish, and I had no knowledge of the reality on the ground. So I think that without them, I would not have been able to get this sample. And I think that this research project is a good example of the value of involving local people in research projects, especially as my results will be of directly used to them. So thank you.
Thank you, Julie. Paige, is there time for a question?
Sure, certainly.
Tina.
Julie, this is a super cool study. I'm so excited for you. Do you have any hypotheses of what to expect between the different levels of anthropogenic stress in terms of the chips or the gene transcription that you're studying?
I'm not sure, but the first part of my project is about genetic diversity of my population. And the first results show that Langlade have really distinct populations. So I hope that it was the same thing with the second part of my research.
Merci.
Okay, thank you again. Up next, we have Javed.
Can you see my slides?
Yes.
Yes.
So today I'm going to talk about regulation of host gene expression by microbiota in Chinook salmon. First thing you might ask, what the hell is microbiota and microbiome? Well, microbiota are a collection of bacteria, viruses, archaea, protists, and algae. And when we study these groups of organisms with their products, that's their genes, in the environment, in a specific environment such as gut, we call them microbiome. When there's association between microbiome and, let's try to use the laser. When there's association between the microbiome and host gene expression pattern, there could be two different scenarios. One is that variation in host gene expression can cause the variation in microbiome. Another scenario would be variation in the microbiome can cause variation in host gene expression. Specifically in this study, by using antibiotic and probiotics, we changed Chinook salmon microbiome and we looked at their gene expression pattern. With this objective that we characterize host response to microbial manipulation, and we had two different hypotheses. One was that we hypothesized that there would be a strong effect of microbiome on host gene expression pattern. And the second hypothesis was that these strong effects would be related to some specific bacterial taxa. So for doing that, we used 2x1 breeding designs. We replicate that 6 times, so that's 12 families, and we replicate that 2, which was 24 tanks. And each tanks there was 5 fish, and we call them 24 tanks, and 24 tanks. These 3 different classes, they received one of them, the first one, they received diet plus antibiotic. In other groups, they received diet, regular diet plus probiotics, and the control groups, they only received diets. Then we collect these fish and we look into first their gene expression pattern using whole transcriptomics. we specifically choose one of the family to decrease the host genetic background. We also collected water microbiome, water samples. We did eDNA extractions, and we look at the water microbiome as well as the gut content. And also we select some of the differentially expressing genes, and we look at those gene expression pattern using OpenArray-CHIP. So as I said, for microbial community, in the next slide, I have more details about it, but we use 16S RNA gene to look into specifically the bacterial community. We use whole transcriptomics to look into host gene expression pattern, and we selected some of the differentially expressed genes, and we select those, and we look into it by using OpenArray-CHIP and included more population. So as you may know, prokaryote ribosome is different than eukaryote ribosome, which has two subunits. One is 50S and the other one is 30S. In 30S subunits, we have 16S rRNA gene. In 16S rRNA gene, as you can see, there is 9 variable region and there are some conserved regions. These conserved regions help us to only amplify the bacterial community, and these variable regions help us to differentiate between the bacterial taxa. For example, it helps us to say whether this is E. coli or Pseudomonas. First, we'll look at the water microbiome. Water microbiome, I mean the water that the fish were living. We collected water from here, we filtered them, and then we did DNA extraction, and we look at the microbial community inside each of these tanks. We saw significant effect based on our treatment. So you can see the color. The green one are the controls. There was a red, and the white one were the red one were probiotics, and there is a white one which were the ones that were receiving antibiotics. As you can see in here, this is a PCOA graph, the first axis and second axis of our PCOA graph. As you can see, each dot is representative of one fish, and the blue ones are the ones that receive antibiotics, fish that receive antibiotics. The red ones are the fish that receive probiotics, and the green ones are the ones that receive the control. As you can see, based on their treatments, they separated nicely and show significant effect of our treatment on water microbiome, which was quite surprising for us because we used the water keep changing. So even a small amount of antibiotics or probiotics can change the water microbiome. In the next level of steps, we look at the effect of our treatment on fish guts, Chinook gut microbiome. The differentiation wasn't that much obvious compared to water microbiome. However, using per-maneuver analysis, we found significant differences between the fish that received. We did pair-wise comparison, and we saw significant effect in fish that receive antibiotics compared to the ones that receive either probiotic or control. And also, we compared the water microbiome with the fish gut microbiome. These are the fish that we collected microbiome from their gut, and these are the water microbiome. As you can see, the water microbiome are separated from the gut microbiome, showing that the fish is actually getting their microbiomes. Other factors can influence the fish microbiome, and if they're not only getting their microbiome from the water. In the next step, we'll look at the host responses to those manipulation. And in here, each dot is representative of a gene, and the green ones are the ones that were more than our threshold for like-fold change, but they weren't significant. The red ones are the ones that were significant and more than the threshold for the like-fold change, and the blue ones were the ones that were significant but not more than our threshold like-fold. We found microbiota depletion by antibiotic mostly lead to downregulation of genes related to immune response and upregulation of apoptotic process. We also found in probiotic treatment genes related to post-translation modification and inflammatory responses were upregulated compared to the controls. Using OpenArray chip, we some of the genes that were significant in here, we looked into them by using OpenArray chip, and we found significant effect of our treatments on some of those genes. In the next step, we used microbial communities, and we saw significant correlation between some of the genes with the some bacterial taxa, specifically Lactobacillus and Bifidobacterium showed significant negative correlation with these two genes. These two genes are related to post-translation modification and immune response, and we found negative association between these two bacterial taxa with those genes. There are different ways that can cause host phenotypic variation. For example, environmental factors can cause host gene variation, and host gene variation can affect the host gene regulation and finally host phenotypic variation. The microbiome either directly can cause host genetic variation, or can cause host phenotypic variation, or as I showed in this study, they can regulate the host gene regulation. They can change the host gene regulation, and through that, it can finally cause host phenotypic variation. To summarize it, we saw antibiotic and probiotic treatment resulted in host and water microbiome. We saw these two treatments caused changes in the microbiome, and we saw manipulation of host microbiome can cause changes in host transcriptome responses. And antibiotic treatment were mostly variation. Fish that receive antibiotic, we saw variation in genes mostly related to immune responses and apoptotic process, while in the probiotic groups, there was more genes related to post-translation modification. This study provides insight into long-lasting question about the ecoevolutionary relation between fish and their microbiome, and also understanding the factor that can affect the fish microbiome and its influence on host health and fitness will help to have a better and sustainable growth for aquaculture. For future direction, first, we tested the first scenario that was variation in the microbiome can cause variation in host gene expression. Further extent of our study could be looking into variation, how variation in host gene expression or host genes can cause variation in the microbiome. It also would be beneficial to look into how the vertical transmission of the microbiome for example, some studies showing that actually parents, that's the maternal and paternal effect of the microbiome. And using knockout and knockdown gene expression would be an extent to this study to, for example, by deleting some of the genes to see if there's association between the microbiome and the host gene expression. I would like to thank my supervisor, Dr. Hiss, and Dr. Chaganti, and also UWindsor funding agencies, GEN-FISH, Ontario Tourism Scholarship, Yellow Island Aquaculture, and Lear for providing me the environment to do this research. If you have any question, I would be happy to answer.
So we have one question from Nicola.
Yes, hello. Very nice presentation, Jawad.
Thank you.
I was wondering, when considering your differentially expressed genes linked to the immune response, did you find some correlation between some particular bacterial strains and some, pro-inflammatory genes such as cytokines or etc. This is something you should go in order to make a more accurate link between the inflammatory status of your fish and the occurrence of strains. And also, it would be interesting to use RNA to do amplicon sequencing instead of DNA in order, you know, to make a better link of activity of your microbiota and the host gene expression.
100% agree, because we use DNA, and some of them, they might be dormant, some of them it might be dead bacteria, but their DNA is there, and we amplify it and we say we saw the bacteria, but they might be actually not present. So I agree with you, including a multi-omics analysis using even metabolites, proteomics, and transcriptomics. Those would be very valid and strong study to include it, and it would be very good extension of this study. I agree with you.
Thank you very much.
Thanks. Phil, you have a question?
Yeah. Can you hear me?
Yeah.
Yep.
Good. Very nice presentation. It was very
Thank
nice.
you.
I saw one of your volcano plots. When you depleted the microbiome, there was a lowering of immune system genes, but there was an increase in apoptotic genes, which I thought was kind of curious because I would have expected the opposite mechanistically. And I'm wondering if you ever looked at like metabolic or certain nutritional pathways, maybe the microbiota are making some kind of metabolite that the cells in the host need in order to survive.
Yeah, so as you said, myself, I was expecting to see more genes related to immune system, for example, cytokines, and one explanation for this would be the controls and the probiotics were healthy fish. So these were having already the good microbiota, even the controls and the probiotics. So one reason would be that we treated with the probiotics for the healthy fish. They had those strains, so we didn't see that much effect on side of the gene expression pattern. But for the probiotics ones, we saw genes were related to apoptotic process and inflammatory responses, which I saw like other studies were reporting the same results. With the apoptotic process, it does make sense, but I also saw like some genes were related to resistance. And one of the things that is hard to compare when there's studies in human and compare those studies with a fish, because their genes have different function and different things. So yeah,
cool. Okay,
I'm not sure if I was able to answer your question.
Yeah, good. Thank you.
Thanks, Javed. Paige, want to introduce the next stop?
Last but certainly not least, we have Ian.
Hello. All right, I will share my screen. Okay, that look good?
Excellent.
Perfect. Okay, hello everybody. My name is Ian Smith. I'm a master's student here at Gleer, and I've been working on a project looking at eDNA concentration analysis and relative abundance in stormwater ponds. So a little bit about the project. I'm sure if anybody's been noticing new residential areas going up around towns, a lot of these places will have these stormwater ponds being built with them. And this seems to be happening at a higher frequency nowadays. These ponds are a way to control stormwater events to prevent flooding in the nearby areas. In the past, these haven't been really seen as natural environments. They've been more looked at as a infrastructure by cities. But because of the progression and the development of natural systems in these stormwater ponds, they're kind of being more seen as a natural environment. But because of these past views, these systems aren't really well understood in terms of their population, and thoughts have arose that these could actually serve as reservoirs or beachheads for invasive species. So what my project is looking for is to develop eDNA as a tool to measure goldfish populations in these ponds, as well as to provide a analysis of a whole relative abundance population assessment. So a little bit about our target species. Goldfish, we all know them. They're a very common pet species around the world. And a lot of these fish can enter these ponds by dumping. While working on this project, we actually found a few ponds that had aquarium gravel scattered around the edge of obvious signs of people dumping these fish. And the problem with these fish is, like carp, they're very hardy fish. They can survive in very low oxygen environments. They have a very generalist diet, and they can survive in very cold temperatures. They also have a very rapid breeding cycle and can very quickly outcompete native species, which is obviously very bad. One of the biggest problems, though, is these aren't really seen as invasive species by the public. Working around these residential areas, people would often comment that, oh, it's, they're nice, pretty goldfish. But recently, and I don't know if this is my biases, there have been many news stories come about kind of addressing the invasive nature of these species. But obviously more work needs to be done to understand how many there are in these ponds and what effects they might have. So these stormwater drainage ponds actually give us a really interesting, unique study system. These are small, almost self-contained environments that have to be regularly cleaned out by a city. For my project, I worked up in Hamilton, and I worked very closely with the Department of Fisheries and Oceans up there, who are also interested in this goldfish problem. And working with the city, we were able to drain 10 ponds over the course of 2 years. And what's very interesting about this is it allows us to actually go in and collect all the fish, including the goldfish, measure them, weigh them to determine biomass, the population abundances, as well as the different species present, which you can't really do for stuff like an ocean or a very large lake or running river. So this is a very interesting opportunity. And we're able to then compare this to any sort of estimates produced by eDNA, as well as estimates that DFO provided via traditional sampling methods. So a little bit about the eDNA concentration that we did. We've done sampling for the last 2 years, 2021, 2022. We sampled many ponds, but before we did any of that, we went in and scouted. We wanted to determine whether the goldfish were in these ponds and to get a good range of ponds that had very high levels of goldfish and very low levels of goldfish. And then some ponds that had very large goldfish and ponds that had very low or very small goldfish, in order to better gauge the relationship between the resultant eDNA concentration that we could take and the actual population types. So these ponds were sampled with a grid pattern. We worked on a 10-meter minimum distance between points, with larger ponds having upwards of 20 samples and the smallest ponds having at least 6 samples. All these samples were filtered on site using an osmos filtering unit, and equipment was decontaminated with 10% bleach solution in between in order to account for any sort of contamination. The eDNA samples were stored in desiccant until they were brought back to Gleer, where I extracted them at the lab. We used the Hernandez primer set, which is at the bottom there. We determined the limits of detection and quantification of this, as well as any non-target amplification of known species present in these ponds. And we found that there was no off-target amplification, which is very useful for the study. So I've been working on this project very closely with Dr. Matthew Yates, and if anybody knows what he does this allometric scaling work with his eDNA and eRNA, and this is incredibly important for my study as well. And what allometric mass scaling is just basically acknowledging that different individuals will produce eDNA at different rates. Smaller fish have a more active metabolism and will produce eDNA more relative to their size than a larger individual. But a larger individual, being larger, will produce more eDNA as it simply will. And so we had to check to see, based off our eDNA samples and concentrations, which or how the ponds should be scaled, whether the numeric abundance of the fish is more important, or if the biomass, the size of the fish, are more important. And all this was done in our studio. As it actually turned out, all the ponds thus far have been completely dominated by abundance and not biomass of the goldfish. And so we got a couple interesting findings. We're not quite there yet. We're still waiting on 1, maybe 2 ponds, a little bit more data. But from what we got so far, we found that there was a very large range of eDNA concentrations through the ponds, which is great. That's exactly what we wanted. We had some ponds that had very little eDNA and some ponds that had a huge amount of eDNA, more than a lot of people have ever seen. A small subset of the ponds had a very high variability. These were ponds that had fish like this, and I speculate that this may be due to some sort of habitat preference, and these fish just depositing eDNA in certain areas of the pond over others. But we did find there was a very strong positive correlation between the eDNA amounts and the population sizes. They're the one. Okay, and the other part of the project is the metabarcoding analysis. So this is the relative abundances of the entire species or the entire population community assessment. So like before, these ponds were scouted ahead of time. We looked for the most diverse ponds for this study. But although unlike the eDNA concentration, these ponds were not drained just due to budget allowances. Otherwise, the eDNA sampling techniques were the exact same. And again, DFO went in and had their own methods to determine their the population assessments. For this part of the metabarcoding, we used the M-MITO and the PS1 primer sets. And because of their prior success in our labs, well locations were marked with the Uni A and Uni B primer sets. The cleaning parameters for the metabarcoding followed prior studies, as well as the sizes of the primers themselves. As you can see, PS1 starts to drop off in quality around 100 or 190, 200. So we went for 190. And M-MITO starts to drop off just after 300. So we went for 290 for the trunk length scores. If anybody's worked with these primer sets before, this is just a breakdown of species positive hits. The green at the top here is unknown or unidentified DNA, and all these individual colors that are not green are positive species hits. PS1, you can see it kind of drops off pretty fairly early, and this graph goes along further. I just had to cut it off for demonstration purposes. M-MITO is the opposite. It was very, very good at detecting many different species, and it does do the same thing with the unidentified DNA. It just does so very far down the list. So on to the metabarcoding results. We found many different species in these ponds, and most of them were what you expect. We found stuff like pumpkin seed, bass, goldfish. There were two species that we found that had a high enough positive hit rate that we weren't expecting. These were northern pike and rough. We did find them to be in pretty low quantities, so this could be just contamination, or perhaps there was maybe a dead individual or something like that. As for the species, they were all detected to the species level except for one. There was a genus level detection of a Pomoxis species, which is some sort of crappie. In one of the ponds, there was a single crappie, and I do suspect, and it did align with the location it was found, so I do suspect it was the black crappie that was found. As for the ponds themselves, they were either dominated by about one or two species, or there was more of a fair balance in between 4 or 5. With examples of pond 27 and 79. Obviously, you can see pond 27 is almost entirely dominated by brook stickleback, and pond 79 is broken up into about 4 species. I'm still awaiting some results on the relative abundance from DFO, but they have provided me at this point presence absence, and a lot of this data does align with the presence absence data. So based off of their presence absence and my relative abundance that I could determine thus far, there's some pretty nice overlap actually. For these tables, the green represents species that both myself and DFO found in these ponds. The white are species that only I detected, and then at the bottom, the orange there are species that I did not detect. So this is pond 27, the graph at the top there. You can see brook stickleback, 95% of the proportional hits from this species. And it does decrease as you go down. One interesting trend that I've seen is that the large or the species with the most amount of hits or a large amount of hits tended to be something that my study and DFO agreed with, and as you went down the list, they kind of dropped off. The same could be true for the other pond that we looked at earlier, pond 79. The top 5 hits were found by both myself and DFO, and as you go down, it drops off. So field applications. There are quite a lot of things that I think the study could be used for. Its eDNA sampling could provide a potential alternative for conventional sampling. Working with DFO, a lot of these small ponds were particularly difficult to sample. They either e-fished them or netted them, and you can imagine a lot of submerged debris or simply difficult access might be difficult for either of those two methods to actually take place or get done in at least a timely manner. In addition, eDNA sampling is a lot less stressful for aquatic species. So in terms of invasive or endangered or rare species, this could be a better alternative. It's more time efficient. We were able to crack out upwards of 5 or 6 ponds in a single day, whereas the DFO traditional sampling could do maybe a pond a day. Maybe it took longer. In addition, the eDNA sampling could tell you a lot more about the pond than simply one style of sampling. And like I was talking about before, this could allow for much more detailed understandings of these endangered or the invasive goldfish species in these ponds. So where do we go from here? I'm not done with my project. There's still quite a few things I want to do. But going forward, I think one of the most important things to be doing would be to replicate the study for low population sizes for the qPCR assays. I focused on goldfish, and a lot of my ponds had very, very high population sizes. So I'd be very interested to see if this could be applied for a low population size so that it could be applied for something like a rare or endangered species. In addition, in being able to compare it to the traditional sampling techniques, a cost comparison analysis would be incredibly important to see if it's even feasible for fisheries managers. In addition, I think there could be some benefit in running a seasonal effect by running it over a course of a seasons. We tried to do that with my study. Unfortunately, we weren't able to repeat on the same pond every single season, and winter was obviously difficult for a number of reasons. DFO has expressed interest in sampling ponds after they've been drained to understand a pattern of succession for the fish species, both native and invasive. And then for my thesis itself, I'm going to be going forward and I'm going to be trying to determine the effect of sampling location within a pond. Fishery managers and biologists may be tempted to just go take a sample at the shore. Obviously, it'd be easier for them, but the depth of the water might interfere with the ability of to make accurate estimates. So for our study, for my study, I'm going to be determining the effects of taking a sample near the shore versus offshore. And the other thing I wish to be doing for this experiment is to run a power analysis to better understand how many samples you actually need to take in order to get a good assessment of population and community composition. Thank you very much.
Thank you, Ian. Are there questions for Ian?
I have a quick, oh no, Brooklyn, go ahead.
Yeah, great presentation, super interesting stuff. So I know just as like a aquatic person, like I'm staring at my 2 fish tanks in front of me right now. There are a lot of subspecies of goldfish. Were you noticing that there was any difficulties with the assay detecting goldfishes or some sort of preference towards one subspecies? Like when I was looking at them, they looked like comet goldfish perhaps to me. But did you notice any trends towards which ones were frequently released or anything like that? Or if there were any sort of differences between which subspecies the assay tended to bind to or maybe didn't bind to?
It's interesting you actually said that, because when we did our sampling and the draining of the ponds, we did find many different subspecies or breeds, or what do you want to call it, of goldfish. We found some that had the big googly eyes. We found some that had the massive tails, some that were orange, some that were almost white, some that were black. However, for this we didn't test for the differences between those sorts of breeds. We assumed that all the goldfish would be treated the same by the actual assay.
Margaret?
Yeah, I was just curious in terms of how long it took to do the metabarcoding part. You said that you could sample about 5 ponds in one day, but of course then there's the lab work required. So either how long it took you, or once things are more routine, what would be sort of the rough timeline between sampling and getting the list of species?
It definitely depends. And for our sampling of a pond, say the 5 to 6 ponds a day, it definitely depended on stuff like the size of the pond and the location of the pond. It would be hard for me to judge exactly how long it would take to get from, say, a single pond to then end result in terms of metabaric co-screening. It would definitely be it would definitely be interesting to do that comparison.
It's in the order of like weeks, a month, like sort of a general sense of.
If I believe if you got a lab up and running, you could probably get it in a week, maybe 2.
Okay,
And if you were able to batch them, you may be able to do a couple ponds at the same time.
Thanks.
Margaret. Hugh?
Hi, sorry, I don't have a screen this morning. I nuked my computer. Ian, occurrence of false positives and false negatives using your eDNA data versus actual fish that were caught in the ponds. Any comments on that?
Oh yeah, so with eDNA, there's definitely a chance of having false positives, detecting things that aren't there. There's also a chance of detecting things that aren't there. As opposed to traditional sampling, you can't detect the fish that's not there. That's one of the benefits that traditional sampling definitely has over eDNA sampling. Although they still have the error present of not detecting species that are there. It's for eDNA, you definitely have to try to strike a balance in terms of how many positive or false positives and how many false negatives you are willing to accept. That you don't have that sort of issue with traditional sampling.
And one addition to that, are you getting the same false positives in different ponds or different species in different ponds? For the false
Can you elaborate
positives.
on that?
False positives, are they always distinctive species that you're getting the false positives with, or do you get multiple false positives in different ponds?
It seems to be different. I wouldn't say there's any particular species that keeps coming up that it looks like a false positive. I will say there could be one that I'm thinking of. I'll go back up to this. As you can see, one species that I detected that DFO did not that has the highest hit rate that didn't find was the black bullhead. And as you can see, what DFO did find was a brown bullhead. I suspect that there may be some sort of either inaccuracy on the assays or the metabar coding, but that also could be an inaccuracy on DFO's point, in which there might have been a misidentification. It is hard to say who got it right, though. I wouldn't be inclined to say that DFO was probably more accurate than the metabar coding, though.
Okay, thank you.
Thanks.
Thanks, Hugh. And Ian, by the way, brown and black bullhead commonly hybridize. And you have mitochondrial markers, so it's a mess.
Yeah.
And I have one quick question for you, Ian.
Mm-hmm.
You said something in the beginning of your talk that I had not thought about. Did DFO get any objections to bumping off thousands of pet goldfish from the people surrounding? Because you can get strong public resistance to culling birds, for example, even if they're invasive. Was there any objections to what they were doing?
I can't say there was any objections, although to be fair, I was mostly there for the eDNA sampling sort of thing. People were very interested in what we were doing. And the few that we did say, oh, we'll be draining the ponds, had seen it before, so they knew that that was a normal thing that happens with these ponds. So there wasn't any major pushback from what I saw, though I wasn't there when they were draining a lot of these ponds. So
Thanks. So Oliver, this is one reason why it's better to do fish, because if they've been bumping off, I don't know, starlings, there would have been trouble.
Anything. You look at a bird wrongly and people get upset at you. So I agree.
Yeah. Thanks very much, Ian. And thanks to all of the 15-minute presenters. And Paige, do you want to start the 3-minute presentations?
Sure thing. Yep, just a reminder when you see your slide to hop on the mic and fire away.
Hi, everyone. I hope you can hear me all right. My name is Riley Beach, and today I'm going to be talking to you briefly about a portion of my master's work that I conducted under the supervision of Dr. Higgs. So as you may know, anthropogenic noise is a sensory pollutant in aquatic ecosystems and has been shown to have negative effects on fish. Generally, the effects of anthropogenic noise are quantified using behavioral, anatomical, or physiological metrics, but at the genetic level, very little is known about noise-specific transcriptional responses. To date, no studies have examined transcriptional changes in fish in the field in response to noise, and very few have examined it in a lab setting. So my study aimed to bridge this gap by conducting a confined field experiment on yellow perch contained in mesh pens in the natural environment. Yellow perch are known to have a mid-level hearing abilities, and boat noise, which is what we used here, spans the entire range of their hearing. So for this study, fish were collected 3 at a time using a seine and either sampled immediately or transferred to experimental pens. Fish were sampled at one of the 3 time points indicated by the arrows on the timeline at the top, which were the following time of capture, the control period, or noise exposure. So fish were either exposed to boat noise or white noise, but no fish went through both treatments as they had to be lethally sampled. So I examined transcriptional changes using the Persiform chip developed for the GEN-FISH group for both general stress response genes and a group of functionally related fitness genes. And by doing so, I was hoping to gain insight onto which biological processes may be associated with noise-induced stress. So the figures here represent the results when genes were grouped by general function, and significant differences were indicated by the asterisks. The only significant differences seen in this study were baseline samples in the stress-related genes shown here, as well as one in the growth and metabolism subset. And with that being said, you can see the noise effects were limited. However, the current experimental design had allowed for the investigation of appropriate acclimation times and potential handling effects. We saw that baseline samples were a little bit more transcriptionally active than the related controls, suggesting that the action of capturing the fish and moving it into an enclosed pen represented a possible stressor. And even though we did not see any noise effects, these results are still helpful as they show the importance of having like a stepwise experimental design, as well as including multiple types of controls so that you can properly tease apart additional effects from those expected from an experimental stressor of interest. And so moving forward in the field, we would benefit from further exploration into noise-induced transcriptional changes, as we've seen that previous studies have shown transcriptional differences in captive fish. Thank you for your time.
Hi, everybody. So my name is Alyssa Weinrow, and I just started as a postdoc with Dr. Ken Jeffries. So I don't have any data for you yet, but it's going to be more of an overview of what I'm going to be working on with him. So I am developing the sea lamprey chip, and as you can see on the side of the screen there, we have a whole host of physiological processes that we targeted those genes from. So we've narrowed down our selection to 56 genes, and I'm currently going through the publicly available sea lamprey genome to pull out those sequences and design probes. We're also using results from a paper that came out of Ken's lab last year, the Lawrence et al. Paper, where they did transcriptomics on lampreys exposed to lampricides. And that's really helped us identify a number of those genes involved in detoxification processes. And so by hitting all of these different physiological endpoints, we're hoping to be able to have a chip that's a bit valuable for a number of different experiments moving forward. And that's all I have for you now. So very short and sweet. Thank you.
All right, hi everyone. I'm Dane Roberts. I'm a master's student in the Pitcher Lab at the University of Windsor. So at last year's AGM, I gave a broader overview of my experimental plans and sort of background, but I wanted to give a bit more of a project update. So, so I work on evaluating strategies to essentially reduce to try and reduce the effects of transport stress in hatchery-reared reintroduced Atlantic salmon. So Atlantic salmon are being reintroduced into Lake Ontario currently via these stocking programs that hinge on transport. So these fish are essentially reared in captivity, and then they're transported to the reintroduction or release site. And during these transport procedures, there's a lot of things that can go on that can stress these fish out. There's oftentimes fluctuations in DO, pH, there's handling during loading and air exposure. A couple different issues that we really want to bring that stress response down prior to that reintroduction or release effort. So that sort of points towards my work. I'm essentially dichotomizing my two experiments into one experiment in which I look at pre-transport mitigation strategies. So what can we do prior to that transport event to bring that stress response down that's induced by transport? So specifically, we're looking at enrichment. And we're also looking at the second experiment, which is on the right-hand side of the screen, which is post-transport. So what can we do after transport? And we look specifically at hard versus soft release comparison to see if soft release is this promising mitigation strategy. So what I've included here, and I can just quickly walk through with the amount of time, is the pre-transport experiment. I have some early descriptive physiological data. So we took non-lethal blood samples after we reared these fish in an enriched environment or a traditional or conventional environment, and for 3 months, and then we transported these fish, and we're comparing these non-lethal blood samples, specifically lactate concentrations. And in our early findings, there's not too much of a difference. It's hard to say at this time, based on these blood samples, if that early enrichment is influencing how the fish are responding to transport. And then if we move to the second experiment, looking at that post-transport mitigation strategy, we did another experiment also taking non-lethal samples of blood with a hard versus soft release comparison. So we followed the ministry to the actual reintroduction release site, and we took blood samples immediately upon arrival at the location, which is considered our hard release fish, and we compared it to our soft release fish. And as you can see, I know it's a little bit small, but you can see the significant reduction in that stress response and lactate concentrations. So you're probably wondering, how does this all tie into GEN-FISH? Reason being is that we took non-lethal gill biopsies throughout these experiments as well, and we're going to run the biopsies on the GEN-FISH STP chip to get an idea of the stress gene regulation going on to essentially fortify and bolster the story to get an idea of whether or not these mitigation strategies are really effective. And the more coherent story that we have, the more likelihood that practitioners will adopt these mitigation strategies and use them in transport procedures nowadays. That's it. Thank you.
Thank you, Dane.
Okay, I'm not a student, but I'm presenting this work because the student that did all the work, Sarah, had disappeared off to med school. So I wanted to update the group on all the hard work she'd done and what she had found. And so this project was designed to look at, do fish habituate and have neural changes in response to long-term stressors, in this case, a 2-cycle boat noise. And it was made possible by donations of the brook trout from Tina's lab. And what we did is once we had the fish from Tina's lab brought in from 2 different strains, we brought them into the lab, tested their immediate responses to a boat noise stressor behaviorally, and then exposed them or didn't expose them for 2 weeks to a constant exposure of this boat noise. And then did the behavioral tests again, did some anatomical tests to see if it was affecting the auditory structures. And then using the brain chip that the group developed, Sarah then ran the genes on the brain chip to see, did we get any up or down regulation of genes we were interested in for neural markers. We did see behavioral changes and anatomical changes suggesting habituation. And so then looking at the neuroplasticity results for the genes, there was 4 housekeeping genes on the chip. We dropped one of them because there's treatment effects. So she then compared everything to a global mean of the 3 of the housekeeping genes. And she found some pretty interesting results on the gene chip. Essentially what she was looking at were the neuroplastic genes shown here. She chose, or found effects of 4 of them. She chose some other ones that showed no effects. And we found upregulation of BDNF, NeuroD1, and SOX2, and a downregulation for neurogenin. Neurogenin is typically a marker for new neuron formation, whereas the other three are typically markers of synaptogenesis and reworked neural pathways. So putting those together, we're not seeing new neurons being formed based on the gene markers, but we are seeing changes in synaptogenesis and neural wiring in response to long-term exposure to this noise stressor. So I think that might be driving some of the habituation things we've seen. There might be some anatomical reasons as well, but at the genetic level, we do see consistent markers for synaptogenesis, not for new neuron formation. We saw a downregulation of stress-related genes, these two highlighted here, the glucocorticoid receptor and the photoreducible RNA binding protein, both were downregulated, suggesting that while it may have initially been a stressor, this habituation effect we've seen at the neural level then act to downregulate the stressor as well. So we're seeing dynamic changes in response to that stressor that might suggest reworking some of the brain neuroarchitecture and might be a good use of the brain chip as we go forward to look at different markers of neurogenesis versus synaptogenesis as markers of neuroplasticity. Thank you.
Do I go now? I can't hear anyone. All right, let's go now. All right.
Yeah, you're good, Adele.
All right. Thank you for attending my presentation. My name is Edel Bai. I'm a student in the Heath Lab. My project is to determine the effects of both triploidy and probiotics on the gut microbiome of Chinook salmon. Triploidy is when an organism has 3 sets of chromosomes. For salmon, this results in them not sexually maturing, preventing any escapees from breeding with their wild counterpart. However, triploid Chinook salmon suffer from very lowered immune function, making them more susceptible to diseases. On the other hand, we can manipulate the gut microbiome directly by giving a fish probiotics, as the microbiome has a great deal of influence on the host's health, possibly negating the lowered immune function of triploid salmon. To test the effects of triploidy and probiotics, we performed a 2x3 cross to create 12 families. We then divide each family into 4 treatments: diploid regular feed, diploid probiotic feed, triploid regular feed, and triploid probiotic feed, with each family and treatment having 2 replicates. We then humanely euthanized the fish and then extracted a microbiome DNA from the hind gut for approximately 280 fish. I then characterized the bacterial community in the fish's intestine by amplifying the next-gen sequencing the V5-V6 region of the 16S ribosomal RNA. From our results, it showed that both triploidy and probiotics seem to significantly alter both the alpha and beta diversity of the gut microbiome, with triploidy lowering the alpha diversity and probiotics increasing it. This shows that triploidy does alter the gut microbiome in a significant manner and could possibly be related to the lowered immune function experienced in triploid fish. Our results had also shown that both probiotic and triploidy had interaction effects with each other, with the differential abundance test showing that diploid had a significant increase in lactobacillus and bifidobacteriaceae due to probiotics. However, in triploid, only an increase in lactobacillus was seen. Another interesting outcome is that the mother has significant effect on the beta diversity of the microbiome as well. This is most likely due to egg-associated microbes from the mother being the first microbes to colonize the gut upon the egg hatching. In closing, the knowledge from this project can be applied to potentially improve triploid aquaculture by negating the downsides of triploidy and help Canadian aquaculture become less environmentally impactful. Thank you for listening to my talk and hope you all have a wonderful day.
Hello, everyone. I'm a postdoc in Ken Jeffries' lab at the University of Manitoba, where we're looking at transcriptional signatures of thermal stress in bull trout. So these bull trout, they're native to the foothills and eastern slopes of the Rocky Mountains in Alberta, and the contraction of their range and population declines over the past century have led to their designation as threatened under the Species at Risk Act and the Alberta Wildlife Act. So increases in water temperature due to climate change, extreme events, and habitat degradation are of particular concern, but we don't actually know that much of their thermal capacity for thermal plasticity or their physiological limits to thermal stress, especially in early developmental periods. So temperatures beyond the thermal range of an animal can elicit a cellular stress response, altering gene expression. So the differential expression of key genes can provide a sensitive indicator of organismal stress before we see any more severe phenotypes like disease or mortality. So we're using the STP chip developed for salmonids by GEN-FISH to measure the transcript abundance of 56 genes related to thermal stress and hypoxia. And on each chip, you can run these genes in duplicate and for 24 individual fish. So one of the many studies that we have on the go is to look at how rearing temperature influences the transcriptional profile and thermal tolerance traits of bull trout during early life. So the eggs and sperm were collected from wild bull trout spawning in Smith Dorian Creek in Alberta. So these adults spawn from August to September, during which time water temperatures can reach up to 8 degrees Celsius. So this is cold for us, but it's actually really warm for bull trout embryos who prefer rearing at 4 degrees Celsius. So if you look at that middle picture, we have these fish rearing at temperature-controlled chambers at 5 different treatments. So we have fixed temperatures of 4 and 8 degrees, as well as fluctuating temperatures from 4 to 6 and 4 to 8 to mimic thermal regimes that they might experience during incubation in the wild. So we're planning on collecting samples at hatch, at swim up, and 6 months after hatch, and we'll be using the salmonid STP chip to determine differences in transcriptional signatures of thermal stress among these different rearing temperatures and whether these persist throughout development. So the hope is to establish relationships between transcriptional profiles and fitness-related phenotypes to identify early indicators of thermal stress at the cellular level to predict any adverse outcomes at the level of the individual or even the population. Thank you.
Hey. Hi, everyone. I'm Patricia Boyer. I am a master's student in the Heath Lab, and today I will be sharing an overview of my project titled Gene Transcription Variation in Response to Environmental Stress Gradients in Freshwater Fishes. Fish encounter a multitude of interacting biotic and abiotic stressors in their environments regularly, and they can either be natural or caused by anthropogenic activity. It is important to understand that environmental stressors are not uniformly distributed across all waterways, but rather they tend to occur over a gradient. And two relevant stressors that we see occurring over gradients are temperature and agricultural nutrient pollution. Now, since gene transcription is the first and rate-limiting step in initiating a gene expression response to environmental stressors, quantification of this process can provide insight to how fish are coping with stressors in their environment. For my master's project, we are setting out to examine whether Canadian freshwater fishes exhibit an adaptive transcriptional response to environmental stressors over a gradient. We started by taking advantage of the Bay of Quinty's natural thermal gradient and a forested to agricultural nutrient pollution gradient along Lac St. Pierre. Additionally, we've expanded our study sites over a larger spatial scale to capture a greater range of temperatures, including several lakes across northern Ontario. We selected yellow perch, tessellated darter, and white sucker as our study species based on their high abundance and because they represent different trophic levels. To observe whether our selected species have either adapted to or are stressed in response to these environmental gradients, we will observe the transcriptional variation among our 3 select species by designing 2 environmental stress transcriptional profiling chips, or STP chips. One chip will be specific to perciform species, which will be fit for the yellow perch and tessellated darter, and the other will be a cypriniforms chip, which will be fit for our white sucker samples. And at this point, I've designed and ordered the perciforms chip, and the cypriniforms chip is currently in the design process. Each chip will include the same set of genes that are involved in thermal stress response and also pollution stress. So genes that are involved in hypoxia, metabolism and detoxification, and also some immune response genes. Using these STP-Chips, we will establish and quantify the threshold at which a stressor will induce a measurable stress response, which will enable us to then predict the point at which these stressors negatively impact fitness in natural ecosystems. In closing, the development of these chips will serve to help monitor fish health in a cost-effective manner, and it will also help us to mitigate potential threats to ecosystems. I also wish to extend my thanks to everyone who is helping with this project. Thanks, everyone.
Am I good to go? Should be. Can you guys hear me? Yeah.
Yep. Go, Nick.
Hello, I'm Nicholas Tugi. I'm a master's student in the Dr. Heath Lab, and my master's is about transcriptional profiling of the osmoregulatory stress response in Chinook salmon. So, in the recent changes of global warming and so forth, there has been an increase of stress response within the environment, and we are designing a OSTP chip. This OSTP chip has a couple of categories: immune function, stress response, growth and metabolism. And with the professors from GEN-FISH, I was able to collaborate which were the most important genes to design on this OSTP chip. And within this OSTP chip, 25 candidate genes and 3 endogenous controls were used. Using a nanofluidic chip will dramatically increase sample thorough output, allowing for shorter analysis time relative to running the samples with individual assays. The OSTP chip will include these functional categories that are directly effective captive rearing performance that are associated with osmoregulatory response. This will be a valuable tool for salmon farming community in general, as there is growing pressure to reduce or ban cage farms, as the fish farm where I worked at, Yale, to collect my samples was actually the same taken down the last couple of years, and I was able to collect both year classes 2019 and 2020 Chinook salmon before they were able to go away. I'll be using gene transcription as a biomarker, as it is very cost effective to do this, and it can produce a lot of data, as once I'm able to analyze the OSTP chips that I just finished running, I will be able to analyze both the control, which would be fresh to fresh water, and then salt to salt water transitions, and then the challenge, which would be fresh to salt and then salt to water. And then we would be able to analyze the osmoregulatory plasticity, as there's an effective need for biomarkers to quantify stress responses, both commercially and conservation related to salmonid rear ending. Characterizing the nature of transcriptic response to such stressors will not only inform captive rearing practices, but also provide insight of the mechanisms of evolution to endogenous species. Transcriptional profiling will give a very great idea of what is going on, which genes are upregulated at specific time points. I have 5 time points: time point 0, 12, 24, 48, and 1 week. And I predict that metabolism and osmoregulatory stress response will be upregulated within the first 24 hours when transferred in between the environments. Now, I also predict the transition between fresh to salt water will increase with a greater osmoregulatory response. And since this is important to understand on why these species are either providing more energy to growing or providing more energy to reproduction, it is important to understand where the energy and allocation is going, as the significance for all of this will be to understand how, as the world is changing and more stressors are into the environment, what is affecting their immune function, their stressors, their growth and metabolism. Ideally, it gives us a slight direction on where their energy and cost is going into reproduction or tissue repair. That is my talk. Thank you for coming.
All right, hi everyone. Last year, I presented some results from our first national survey paper, which we did as part of Activity 5, and we have a second one on the way, but it's not ready yet. So in the meantime, I'm going to present some work that I did this past year on survey methods. So when we approach research questions like ours, we have two choices. If we're doing something like a survey or a focus group, when it comes to sampling people, we can sample probabilistically from a group, meaning we sample randomly and across relevant strata from a known population, or we can go non-probabilistic, meaning potentially we sample snowball style from a population that has no sampling frame. The full-scale probabilistic survey might provide us with statistically rigorous data, but at a high cost in terms of time, money, and those data might have too low a resolution to answer certain questions. The non-probabilistic survey, on the other hand, is fast and frugal, but these data are bias prone, and in general, we don't know how biased these data are or in what ways. So this begs the question, what can we actually learn from biased data? To try and answer this, I reviewed literature in the human dimensions of fisheries, as well as non-fisheries literature on non-probabilistic methods. And a lot of the recent research in this space deals with bias mitigation and correction strategies, things like demographic quotas and weighting, quasi-correcting biases in data post hoc. The problem with these methods is that while they are improving, and remarkably so in disciplines like health sciences and marketing, they're still fundamentally limited, meaning we can't render generalizable data from just any non-probabilistic sample. And I think the simplest and most appropriate response to this problem, at least at this point, is to ask appropriate questions and play to the methodological strengths of non-probabilistic methods, if that's what we're going to use. There's lots of research in the human dimensions of fisheries that does this, and there's lots of insightful research as a result of that. But there's also some of what I would call awkward surveys or, focus groups or other research methods that these are cases where the research questions and objectives don't line up perfectly with the research methods. For example, a non-probabilistic survey targeting population inferences at a large scale. But it doesn't have to be this way. We can set up our research questions and our studies so that they're more insightful and that they have higher inferential power than this. One way that we can do this is to provide supplementary data in very niche but important areas. And a good example of this in the human dimensions of fisheries is to provide data on highly specialized, avid recreational fishers. These are groups that are impactful and important, but are missed in large-scale surveys. And one way that we can use non-probabilistic data supplementarily would be to gather information on these groups and, let's say, use it to boost a model that's fitted to probabilistic data and provide understanding that's relevant to fisheries managers. Another example of how we can play to these strengths, and this is the approach that we took with our research in Activity 5, is to zoom in on niche groups in phenomena. Like in our case, we chose a hard-to-reach group in freshwater fisheries managers, and we chose some complex phenomena in science policy interactions. And this allowed us to ask questions like, why doesn't scientific progress lead to proportionate progress in management and conservation? And more importantly, can we improve this? So our first paper, which again I presented last year, provided some answers here, and there's more to come in paper 2. The takeaway message is that we're learning valuable lessons from biased data, but they're the right kind of biased data. They're biased toward the exact kind of expertise and insight that we're looking for in Activity 5. And there's no shortage of opportunity to do this in the human dimensions of fisheries. We just need to, again, understand the methodological strengths of whatever approach we're taking and then research accordingly. Thanks.
So another huge thank you and round of applause for all of our HQP and student presenters. I think it goes without saying these projects are some of the most exciting work coming out of the GEN-FISH project. And I know we're all really grateful that you guys took the time to prepare and share your research. Are there any brief questions at this point before we take a break? All right, I'm not seeing anything at the moment. So at this point, we will step away for a brief break and meet back here at around 1:30 Eastern for our co-production workshop. Thank you all.
Thanks, everybody.
Hey, Paige and Dan, I'm showing 132. Do you want me to forge ahead?
Yep, I'd go ahead, Steve.
Excellent. All right. Hey, folks. Good to see everybody. For those that don't know, my name is Stephen Cook, and I'm one of the PIs for the GEN-FISH project. However, we have some new folks on board. We have some visitors from the NSERC Crate Fishcast program. We are offering this as a micro-credential for the students that are in that program as well. They will be joining us again in January for an additional session that's a bit more interactive. But for today's purpose, today's workshop is more on the Steve gets to pontificate side of things. It is me spewing information at you. We have shared some resources, and there's some additional resources that sort of get shared throughout this this presentation. And certainly, if you have any questions for me afterwards, I'll throw my I can't talk and type at the same time. I will throw my email into the into the chat there at the end. You're welcome to reach out to me or Vivian at any point in time. You'll notice that Professor Nguyen is not here. That is because she is teaching an undergrad class at the same time. So I'm running solo today. Vivian is a scholar who works in the area of knowledge mobilization and co-production. I'm more of a, I've learned by doing, both through successes and failures, and then Vivian's been able to put words around what I've done and help me understand why I have failed and why I have succeeded. So we work quite well together as a team, and I think that's what you will see today as we work through this. It's a combination of material that's anchored in the literature and then some of our own experiences. And I'll use three brief case studies towards the end. So we've got a little less than an hour to get through this. Before we begin, and I have to hide all of you, so I'm not going to be able to keep my eye on the chat. I strongly encourage you to use the chat throughout. There's a number of other folks on this call that do a lot of co-production work. I'm looking at Tina, for example. I would love to, at the end, view the full screen and see a very lively chat record there. So feel free to share resources, ideas, perspectives. But while I still can see the chat, I'd like to get an idea of the folks that are on the call here. If you have done any work in the co-production space, or alternatively, if you're on the call saying, what is co-production? So can you let me know where you are, sort of, or maybe somewhere in the middle? So if you've done something, yes, no, a little, just trying to get a feel for folks. Throw something in the chat if you've you're welcome to put any details too, like, oh, did it on a project on, with an aquaculture organization or this or that. Honestly, don't know what co-production is. Excellent. We'll see whether at the end, whether that's changed, hopefully. Currently co-producing PhD. Perfect. Yes. Seeing some thumbs up. I did. So a good mix. And so I welcome, if I can get through this, I certainly welcome at the end folks sharing their own experiences. I'll talk to Tina as well. Maybe we're able to open up that the January event to those that want to dig a little bit deeper, but that aren't Fishcast students. Can't give you a credential, but we can still have you be part of that conversation. Okay, so with that, I'm going to hide all of you. So I'm not going to be able to see any of you. So you can, it doesn't matter whether you keep your camera on or off. I'm going to blank that. I'm going to move that. Okay. Because of this, if something goes sideways and all of a sudden I've lost connection, somebody's going to need to let me know, because all I see is my screen. And so with that, let's talk co-production in the environmental sciences. So a lot of this started for me, this idea of trying to be more effective and relevant as an applied researcher out of frustration. And that frustration really has sort of been simmering for more than a decade. Some of that is reflected in a paper I wrote with a number of students that were in my lab. You can see it there, being relevant. There's a few places where I include the little QR code. So if you want to scan that, it'll take you to the paper. It's open access. But we started to really reflect on, how do we, if we're going to do applied research, then it should either have impact or have the potential to have impact. And I was pretty frustrated. The first part of my career, I was being a good academic. I was publishing papers. I was doing work on applied topics, but at the same time, I wasn't seeing that impact on policy and practice that I expected. And I've since learned there's all sorts of reasons for that, and they're very complex, and co-production could not have solved all of those issues. But nonetheless, it's part of the equation. And I also realize I have yet to define co-production, and I'm not going to for a few slides here. By way of introduction, I also want to share that I'm going to speak largely from the experiences that I've had, and most of the co-production work that I've done is with practitioners, policymakers, and that work for parliamentary governments, as well as anglers and commercial fishers. I've done relatively little work with Indigenous communities that I would be proud of from a co-production perspective. We've done some, and I will share a few examples, but there are a variety of reasons why it's very challenging to do that, and it's something that we've struggled a little bit with as GEN-FISH because it takes so much time. And the challenge I find is that time may not align with a grad student tenure in our lab. And so, and as somebody who's supposed to be training grad students, that's a bit challenging. And so you have to establish long-term relationships and work on things in that way. So we're working on it, but there's some room for improvement there. So I just wanted to share that as a background. Okay, so there is this thing called the knowledge action gap. It has other names that you can see there as well. And this is the idea that there's all sorts of science or knowledge over here, and there's all sorts of people that want to apply it over here, and there's a gap between. And so a lot of work is spent on trying to build bridges. How do we connect knowledge to action? And social science work done by people like Vivian, Carly Cook, and others has told us that the ways in which knowledge is used or ignored in decision making, and then of course we have our own experiences to bring to the table. There's certainly lots of barriers to the uptake of new knowledge. Research is showing that complex knowledge in particular does not travel well. It used to be assumed that knowledge moved in a pipeline model. So at one end is you, the grad student, and you're pumping out papers and you shove them into the pipeline. And that could be an email. You're emailing them to a practitioner, or you're just publishing them. And that's just assumes that they're just waiting at the other end of the pipe, ready to receive that information. We now know that knowledge moves in really complex ways through networks. It changes, it morphs, and that influences the extent to which it actually yields tangible changes in policy and practice. All of this fits within this broader lens of knowledge mobilization. And so let's start in the middle there. And that's Vivian in the corner, by the way, if you don't know Vivian. This is a schematic that came out of her PhD, and I keep going back and leaning on it because I like it quite a bit. In the middle, we've got what we're calling the knowledge mediation sphere. And so that's sort of this middle ground between, on the left side, those that hold or generate knowledge. So think researchers, but it can be certainly others as well. And then on the other side is sort of that application or action side. So those are decision makers and so on. These systems do interact through this knowledge network that this domain that sits between the two. And the idea of co-production, which you can see on the left there, is that we're able to bring these spheres together in a meaningful way. And I will describe how we do that in a moment. I also want to point out when we do social science surveys, so this is work from Nathan Young and Vivian, and we ask stakeholders and fisheries managers in British Columbia, what are the barriers to incorporating new science into actual fisheries management practice? And you can see there's differences between the stakeholders and managers, but I've highlighted ones in red that are particularly important and things we can address with co-production. So one is trust issues, and we see that the word trust appears on both sides. The word transparency appears on the right. I would argue transparency is very important for stakeholders as well, even though that did not appear in our survey. And then also the relevance and reliability of findings. And so you kind of drill down and think about what does it mean to be relevant? What makes things reliable? We can also look at individual researchers, and Vivian did a really cool survey of people that are active in telemetry, fish telemetry tracking studies that engage in applied research, which is frankly 99% of the people that do fish telemetry. It's to understand habitat use, it's to assess mortality. So these are pretty fundamental things to fisheries management. And we asked those individuals about the work they've done, the extent to which in their minds their work has been impactful, and the characteristics associated with that. And there were a couple of things that came out of it. One was that people that were generally altruistic, so researchers that express sentiments that suggest they were altruistic, collaborative, and engaged in pro-engagement behaviors, that was positively associated with the successful uptake of findings. And then we also learned that researchers that were involved in fisheries management processes experienced more successful knowledge outcomes. And so those are, for example, stock assessment biologists that work at that interface between research and management, government research scientists that are embedded in management, or academics that work in the management sphere, for example, sitting on committees with the Great Lakes Fishery Commission. So we know that when we put scientists and managers together, we get some better outcomes. All of that can be wrapped up in a phrase called knowledge co-production. And that's research that's conducted collaboratively, inclusively, and in a respectful, engaged manner. And it extends from identification of the research needs. So what are we going to do? How are we going to do it? Actually collecting the data, interpreting it, and then on the application side. And the idea is that this creates actionable science and benefits all of the partners involved. So the idea here, again, the status quo is scientists do science. Whether they're a government scientist, whether they're an academic scientist, scientists do science. And co-production is a little bit different. It's recognizing that maybe we should actually bring the practitioners into the tent. Maybe we should bring the Indigenous elders and Indigenous knowledge holders and Indigenous resource managers into the tent. Maybe we should bring the frontline fisheries managers into the tent. And we can also do that with communities, whether they're Indigenous communities or not. We can do that with specific stakeholder groups like commercial fishers, recreational fishers, and so on. And the idea is that you work together collaboratively. And I talk to a lot of people and ask them what co-production is, or if they do co-production. They say, sure, I do. At the very end, after I've done my study, I go to the community and I do a presentation, or I send an email off to fisheries managers, or I do a presentation for fisheries managers. That's nice and all. That's called science communication. But knowledge production begins at the front end, and it's a process, and it extends through time, and it leads to continual interaction and engagement. And what we see is knowledge exchange. And I'm using that word very purposefully instead of science communication. Oftentimes we think of science communication as what I'm doing. Again, I've got the floor right now, and I'm just chucking information at you. We know that in terms of knowledge exchange, what's more productive is the give and the take. You listen, you learn, you share, they listen, they learn, you share. So we're exchanging ideas and perspectives and so on. And anybody that's been in a relationship, a successful relationship or a failed one, I'm talking about a personal relationship, you know how important that is. It's not just communication. It goes in both directions. It needs to be that for it to be an effective relationship. So this idea isn't fringe by any means. The journal Nature did a whole special issue on it in October of 2018. There's some really cool papers in there, including some of these that are more news items written around it. And at some point along the way, I guess it was right when the pandemic was about to hit, gathered a bunch of folks when we were sitting at home in our undies, and we wrote a paper on knowledge co-production, very much focused on the fisheries science and management communities. We are not the first ones to talk about co-production. We were just bringing it to that community in using examples that we thought would resonate with them. And so much of what I'm going to share today spun off from that paper, and that paper was a GEN-FISH product, although it also looped in folks from 2 other Genome Canada projects so that we had a greater diversity of ideas. So before we move on further, I want everybody to just take a minute and think about, what does success mean to you as an applied environmental researcher? And whether you are or aren't, I'm going to paint you with that brush because that's what Fishcast does. That's what GEN-FISH does. So what is success? Is it publications? Is it students graduated? Is it getting invited to conferences? Is it awards? Or is it changes in how we actually manage a fishery? Is it saving dollars in how we approach management so that we can ensure that the limited resources we do have are invested wisely? And we're certainly learning now that how one does applied research is as important as the outputs. If you do your work in a bad way, and I don't mean in terms of fudging data, I mean in terms of not engaging relevant actors and engaging in things like co-production, we see that work tends to be more and more ignored. And so I'm just going to play on this next slide the start of a video. I'm not going to play the whole thing, but this is a little video we put together that spins off of that paper I just showed. So I'm going to see if this works.
Let's talk about success in applied environmental research. What is it? How can it be achieved? And how do you know when you have achieved it? First, let's define what we mean by success. Success in applied environmental science is respectfully conducted, partner-relevant research that is accessible, understandable, and shared with the potential to create an opportunity for change. Given that resources for conducting research are limited, applied.
Did that work?
We heard the volume, so we heard it, but we couldn't see it. And so I think Connor was saying that maybe it was playing behind in another window.
Probably. Okay. All right. I'm just going to skip this, but I'll send everybody the video link. If you heard it, that's what I care about most. And in particular, you would have heard about a definition that we came up with as a group as to what success meant to us. And it was something that we worked on quite a bit, and it took a while to find consensus on. But co-production was part of that. And the reason is because there's a large body of research that shows that co-produced research is research that is more likely to be embraced by end users and other partners. So now it's going to play. Okay. Before we move on and really talk more about co-production, I need to introduce a few other terms. One is knowledge co-assessment. And so this is so Bill Sutherland and team argued a few years ago that much of what is and need to be doing isn't co-production, but co-assessment. co-assessment is when the knowledge already exists. So let's say we were doing a workshop and we wanted to bring in experts around a topic such as, stream restoration. What's best practice for stream restoration? And the idea is you assemble what's known in the literature, you bring in experts. Those experts do not have to be scientists. They might be stewardship groups, they might be practitioners, and you sit down and you work through the process together to assess what's known and determine the extent to which you're able to sort of bring that knowledge to bear on decisions in a useful manner. And then the idea is that co-production is only necessary if there is a knowledge deficit or if it's not relevant to the work that's being done. So I think this approach is relevant, and that's a reasonable label for work that is more synthesis oriented as opposed to work that is focused on generating new knowledge. So that's knowledge co-assessment. Another term is knowledge co-evolution, and this has been brought to the table in the context of working with Indigenous communities. And indeed, it's an approach that's not unlike Two Eyed Seeing. And the idea of co-evolution is that, the problem with knowledge co-production implies that you can only produce something new. And the reality is that if you're working with Indigenous knowledge holders, they may be bringing generations of knowledge to the table. And although the idea is to bring those, to look at those through both lenses, to try and bridge those systems, the one exists. So it's how do you interpret these things in the context of each other? And so that's essentially this idea of knowledge co-evolution, that both knowledge systems have the potential to advance and evolve on their own, but also come together where relevant. And specifically here, they note this is about generating new knowledge for the benefit of Indigenous stakeholders. This is a fantastic paper. Is written by Jacqueline Chapman, who was a PhD student in my lab when she did this, but she did 2 PhDs. She only got credit for one, the one she did with me, but she hung out a whole lot with Stefan Schott, as well as Louis Bernatchez and others working on a project called Fishes in the Arctic. It was a Genome Canada project. And this is an incredible paper that they published. So if you're working with Indigenous communities, thinking about knowledge co-evolution is certainly worthwhile. It's a relatively new concept. It's something that I have not had the opportunity to fully engage in. So sharing that as a learning opportunity. Okay, so I'm going to come back to co-production now. So why? What are the benefits? And I've told you a few already. One of the biggies is that it changes the way or advances the way in which science is viewed and valued by different actors. And this allows science to be incorporated into natural resource management. And this can be through increasing the understanding of different user groups or actor groups, helping practitioners understand science and value science. It also provides an opportunity for scientists to benefit from that broader perspective and understanding of the impact of their work and how that work can impact people and their decision-making processes, which can feed back and again change maybe how we approach our empirical research, even the work that's not done, even work that's done outside of a co-production model. It reduces wasted resources, because if you've taken on a co-production model and say it's with the idea is that you're supposed to be doing work to help better manage the commercial fishery on Lake Erie, well, you're going to be working very closely with fisheries managers and decision makers that will tell you whether or not the question that you're answering is a good one. They will tell you the questions they need answered. They will be involved in designing the study and executing the study, interpreting it. So it ensures that at the end of the day, they're getting something that they need and that they can use. It also provides an opportunity to empower individuals and groups that have been traditionally marginalized and left out of the scientific process. It can be used to address conflict because it puts people together to work together on a common problem, on a common issue, and that leads to understanding, and generally creates more inclusive processes so that we get better outcomes. It also increases the relevance and applicability, which we've mentioned already. We know that empirical social science tells us that that co-production is perhaps the biggest single thing that a researcher can do to help bridge the knowledge-action divide. There's many things one can do, but if one embraces a co-production approach, that requires so many of those things that we know about relationship building, coming together, being a good listener, working collaboratively. All of that is embedded within co-production. And so if you engage in co-production, we really see that bridge narrowed between knowledge and action. I'm going to give you some examples later on. It disrupts that unidirectional, linear, and sort of isolated approach to research where researchers over here and knowledge users over here. So it helps to bring people closer together. And it really creates opportunities for increasing interpersonal trust. And I showed you on a previous slide that trust is important for both stakeholders and decision makers when it comes to using science. And if they trust the people in the room, they trust the science that's done, that's more likely to lead to their work your work actually being embraced. And then finally, working alongside knowledge users throughout the whole process really makes sure that you keep your eye on the ball, that you remain relevant, that you don't end up going down rabbit holes. It promotes credibility, saliency, and it brings legitimacy to the work done. So lots of benefits to co-production, but it ain't easy. There are a number of challenges, but fortunately, most of these can be overcome. The first challenge in particular is one that I don't have much concern over. This idea that it impinges on your ability, your freedom as a scientist to be fully independent and do the work that you want to do and you think needs to be done. Because co-production inherently is more of a democratic process. And you may think that this is the direction that you want to go. And your end users may say, that's not what we need. This is what we need. And you can choose to ignore them, or you can choose to work with them and give them what they truly need. So this is one that I'm not concerned about as long as that process starts early. You don't want to have a grad student that's 2/3 through their project and all of a sudden things are changing, but that's not co-production if that's the way you're doing it. It does require specific skills and competencies. I'm not going to pretend today is a full-on training session. We're working to build some competencies and understanding, but there's certainly more work that can be done. And really, there aren't very many formal opportunities to do this. So most of us learn through trial and error and increasingly leaning on others that have had successes and failures in this space. It can be costly in terms of time and funding to develop relationships. It's a long-term investment. Much of this is slow science. And in fact, it may take years before any science actually takes place. It can also be unexpectedly costly to the knowledge users, stakeholders, and rights holders. Working with Nathan Young, we've done some work on the pressure and stress that's borne by Indigenous community leaders and elders who are repeatedly pulled upon to represent their community on all sorts of issues related to healthcare, education, the environment, fisheries. And so they have it's a lot of stress on their shoulders to try to represent their community views and to bring that intergenerational knowledge to bear. So it's costly to them. It's difficult to ensure that power is shared. You know, if you're the researcher and you've got the money to do the research, you know, at the end of the day, you're the one that's going to be judged on it. It's challenging to cede some of that power and really approach it in a way that all participants have a voice and are respected and encouraged. And then finally, on this slide, this idea that Indigenous knowledge, so this is specific to Indigenous knowledge, although frankly, it's it's difficult to, most knowledge systems, I think this is relevant to. Indigenous knowledge is inseparable from sociocultural, political, and legal values that give rise to our systems. And it may, especially when engaging with Indigenous communities, we need to move away from this idea of knowledge extraction to collaboration and partnership. And that's something that requires thinking about things like OCAP and certainly going about things in a really purposeful and good way. So you've decided that you want to embark on this path. So where do you start? So we're going to start with the red fish there, and we're going to swim in a circle. That's identifying potential partners. Sometimes this comes easy. The phone rings, a knock at the door. It's a community. It's a manager saying, I got a problem. Can we work together on this? Other times it's more researcher driven, where it's like, hey, NSERC's got a call to do work on effects of microplastics in aquatic systems. I'm interested in how that might affect communities. I wonder if communities are also interested in that. I'm going to make some phone calls. I'm going to try, and so you start to ask around, start to build relationships. Then we move on to that assessment stage. So this is where that co-assessment could fit in, saying, well, what do we already know about this problem? What do we think about this problem? Where are those gaps? And then working collaboratively to plan the research, generate the new knowledge, communicate the findings, although in many ways that happens inherently because they're part of the process, applying the knowledge, and then it can sort of go in a bit of a circular phase. Of course, there's feedbacks and loops that can sort of spin off this. But in a general sense, these are the typical stages. There is some general guidance that we can provide for doing co-production, but there's nothing that I can guarantee will work in every scenario. There's a lot of subtlety here, and a lot of it depends on the partners and their ability, interest to engage, and about that relationship, the depth of that relationship and trust. And you need to build that before you can really get too deep into this. So just remember that. Not all of this is relevant to every case. Here are a couple key resources. I'm not an author on any of these, nor is Vivian. These are what I think are some of the classics from other, I don't want to say disciplines, it's just less fishy, more broadly in the conservation and environmental space. So all good papers, all worth checking out, and there's certainly others as well. So I'm just dragging on my talking here so people can screen capture those. So, okay. All right, so first one. We mentioned this already. Build strong and lasting relationships with partners. That really is a prerequisite for co-production. And as I've said, it takes time and effort. It doesn't happen overnight. A partnership is not having one phone call and saying, hey, do you want to work together on this? Sure. That might be like indicating interest, but that's still not a partnership. You really have to get to know folks and their interests quite well. Before engaging in co-production, you also need to sort of come up with some guiding principles to make sure that everybody's on the same page. What are we trying to accomplish here? How are we going to get there? What type of interaction are we going to have? When is that interaction going to occur? How do we ensure that everybody has voice, and so on? And that may be as formal as some kind of legal document, or it may just be a conversation saying, hey, let's agree to work together and do this in a way where we try and reach mutual benefit, and at the end of the day, solve a problem. That, of course, involves discussing the goals and perspectives of all involved, and that's to understand the context. Everybody has different cultural, institutional, socioeconomic context from their value systems, from their lived experiences, from their training, that will influence how they engage and what they consider to be the goals and sort of objectives of a given project. Another really important part, especially when engaging with Indigenous communities, is really explicit and early discussions about power sharing, responsibility, and ownership. if Indigenous community members are going to be sharing their perspectives, and in particular, if they're sharing intergenerational knowledge, then there's expectations. Fortunately, the OCAP principles can help guide us, but even coming up with an agreement on how to implement the OCAP principles, that can take weeks, months, even years, depending on how delicate a file is, depending on where a community might be in treaty negotiations. There's all sorts of things going on behind the scenes, and a heck of a lot of history that it takes time to understand as a settler. And that's going to vary from community to community, from Indigenous government to Indigenous government. There is no one way, there's no one path. One of the keys here, and I've said it and I'll keep saying it, is you got to start early, ideally on that problem identification. Hey, what are your issues? What are the problems that are plaguing you, and and how can I help? And as a researcher that does applied research, I think that's really, really important. How can I help? Right? If we're trying to reverse the freshwater biodiversity crisis, the focus of GEN-FISH and FishCast, then with maybe even a slightly even fishier focus, then we certainly want to be listening to fisheries managers. We want to be listening to anglers. We want to be listening to Indigenous resource managers, environmental consultants, are the people that are interacting with and that depend upon these resources for livelihoods. They have a legal responsibility to do this kind of work, for nutritional security and cultural exercising cultural rights and so on. So start early. Mentioned this already, this idea of sustained bidirectional communication, or what we call knowledge exchange, making sure that it's not you as the all-knowing scientist on one side simply sharing all of that wisdom that you've learned from all of those books and journal articles that you've read. When you sit down with partners, it's a gift. It's an opportunity to learn from them about how the world works through their lenses, their training, their lived experiences, the hats they wear in their job or lives. So, as I've said, it ain't easy. And there are people that are gifted in this space. In particular, there's individuals that identify as being knowledge brokers. And there is training you can take. Guelph has a good knowledge brokering program. And there's a knowledge mobilization, knowledge brokering program associated with the Canadian Science Policy Conference, which is every year, led by some folks out of Ottawa, and I think they do some free work as well. So this is another potential job option for those of you in the sciences. So a knowledge broker is somebody that's sort of an intermediary. They're not a practitioner. They're not a knowledge generator. They may have had experience in both and training in both, but their job is to be in that middle space, to help facilitate co-production, to facilitate relationship building, to facilitate knowledge exchange, and so on. So NSERC is allowing the inclusion of this in grants now. So if I'm doing applied research and I want to include some money for a knowledge broker to help with this kind of work, that's entirely a legitimate expense. Certainly at the core of all of this, when we talk about relationships, is this culture of respect. If there's no respect, there will be no trust, there will be no progress, there will be no sharing of meaningful perspectives. There will likely be conflict if people engage at all. And so you can see some of the words around the edge: civility, authenticity, equity, inclusivity, diversity, that all feed into thinking about that culture of respect, and ensuring that everyone's voice is given equal importance. As one does co-production, you can imagine that it's not entirely linear, and that there are lots of feedbacks and opportunity to reflect and refine. And in fact, I'm going to give you some examples of that a little later on. And you should do that reflection with the partners and stakeholders. So, hey, how'd that phase go? What did we learn there? Okay, moving forward, what do we want to change for the next phase? And if we start from the beginning again, what would we do differently? So just taking the time to reflect and refine as needed. Certainly, this is something, if we're going to be working with Indigenous community members or other individuals that bring unique knowledge to the table, we need to think about what's culturally relevant, appropriate, and create safe spaces so that Indigenous research can also flourish, or community science, and fit within existing knowledge production systems. I think there's still this idea that there's the wisdom held by Indigenous peoples, and then there's this new science we do. Indigenous knowledge is a form of science, and Indigenous community members and natural resource managers and scholars also do Indigenous science. So there's space for both bringing in the traditional knowledge along with new knowledge, and making space for that. And then I'm not going to rehash this. This was a figure in our fisheries paper, but you can see there are a couple key things here that all fit into various forms of the guidance I just shared. This is what we thought were the most important: sharing of power, being inclusive, creating a space where there's equality, reciprocity, and the importance of building and maintaining relationships. Okay. So I'm going to work through a few examples here. And the first example comes from Florida. And this is work that's been led on the research side by Jake Brownscombe. And Jake did his postdoc working on permit. And we now have there's a PhD student that he and I now co-supervise that's moving forward in this space. And this was an example of a project where Bonefish and Tarpon Trust, an NGO with interest in bonefish, tarpon, and permit, even though permit's not in their name, came to us and said, hey, we have some science needs around this, and we're wondering if you could put together a proposal. And so that's an obvious place. We're like, okay, this is new to us. Who should we engage with? Who do you And so they created an opportunity for us to connect with resource managers, fishing guides, and other experts. And very early on, we worked collaboratively to figure out what that proposal would look like. And it became clear there was a fair amount of concern around fishing at spawning aggregations in the spring. There was a seasonal reef tract closure in place. That was based on gonadosomatic index studies, and that they were using that to decide when to open and close these areas. And so there was interest in the community to use acoustic telemetry to track fish. And so Jake, who's in that photo there, worked with fishing guides. So he had to build the trust such that they basically trained him to become a guide. Gave away some of their best spots, allowed him to work alongside their clients so that they could get permit tagged. That took time. In the first year, we tagged 1 permit. In the second year, we tagged over 30. And that was all about relationship building and knowledge exchange and learning from those local experts. And so what happens is permits spend a lot of time in the shallow reef area, the shallow mangrove flats. And then in the springtime, they move off to these reef tracks or wrecks along the deeper water there. You can see circled in yellow. And it turns out that they move out a lot earlier. So the black bars are when we detected permit at the spawning grounds. And you can see the green there is the period at which harvest was prohibited. And so there was a harvest-oriented fishery that occurred that was legal during March and particularly April, that allowed folks to hammer some of those fish that were already at those spawning aggregations. And so using the data that Jake collected, that date was advanced. And it happened remarkably quickly, okay? 2011 is when that initial project, that initial closure was put in place. 2016 is when we started the telemetry study. You can see there in 2017, the Florida Fish and Wildlife Commission formally requested information on the summary of information available. We did our first download the following year, delivered reports, and then lots of back and forth with the commission, and some of their scientists and managers were working with it. A draft rule was approved in December 2017, and in February 2018, the legislation passed, and there's now a May to July special permit zone to protect these fish. That is written up in this paper because of how rapidly it worked. And the reason that it was so fast is because the government agent, first of all, we were doing work that was relevant. It was information that they needed. Government agency partners were actively involved in the research, and some of them were co-authors in the previous page. The methods were well accepted by both government and the stakeholders. And so there's one of the fishing guides off to the right. And you can see there, and what that did is very early on, getting them comfortable with the fact that we were tagging animals, that this was generating useful data. There's funding and lobbying activities that BTT engaged in. Anglers and guides are actually involved in the research, and widespread support among the guides. You can see down at the bottom there an example of some of the work done with donors. You could basically adopt a tag, adopt a fish. So anyways, this is an example of co-production that happened remarkably quickly and yielded change remarkably quickly. If we just did our academic thing and operated in a vacuum, I'm pretty sure that legislation would not have changed. We probably still would not have tagged more than a handful of permit because we depended so much on those fishing guides. So one example. Another recreational fishing example comes out of the muskellunge space. So the fish of 10,000 casts, a highly specialized fishery. Today, many of those fish are released, somewhere around 99%, but they historically were subject to overharvest, and you can see a picture there. Lots of initiatives. The Muskies Canada and the community has long been involved in doing angler diary programs, promoting proper handling, funding research, and so on. Working, that's Sean Landsman in the bottom left. He did his master's on a Muskies Canada project, and they came to us and said, we're concerned about catch and release. We want to make sure that we're not impacting these animals in a negative way. What are the best practices? And so we said, sure, we can do that work. And so we worked with them to develop specifically what were the questions that fed into what the experimental design was. And because these are the fish of 10,000 casts, it takes a certain kind of field assistant. And that's Hedrick Wachelka, who's the head of conservation for Muskies Canada Ottawa chapter. He's retired, and he was Sean's field assistant. And when Hedrick couldn't be there, he coordinated other Muskies Canada volunteers. And so that co-creation or production of the research agenda, we had avid anglers, highly specialized. They co-funded the work. They were involved in the capture and tracking, and it was constant communication in the boat together at meetings. And so this led to updates in their practices and in their educational materials. And here's just an example of some of the communication work that Sean did, in addition to the direct knowledge exchange with the core group. It was also trying to have a broader reach through magazines, a blog, newsletters, and so on. The last example comes from Toronto Harbor, and this one's a bit different. This is a project, what I'm going to describe is based off a paper led by Morgan Pixak, and that was published last year. And this is focused around a co-production relationship that's mediated by a group called Aquatic Habitat Toronto. It is an entity. There's no building. There's no it's just a group of people that come together, and you can see a long list of names, and you're not going to recognize many of them from the literature because the majority of them are practitioners that work for different organizations. So a lot of restoration work going on in Toronto Harbor, and so making sure that the work's being done in a good way. So Aquatic Habitat Toronto is a consensus-based partnership that undertakes knowledge co-production. So collectively, the group, and you can see there, it's everything from regional governments, provincial, federal, local, or regional, Indigenous community, and several universities, and we get together, we talk about what the science needs are, the management needs, that leads to the science needs, then there's funding, we do the work, gets shared and interpreted through Aquatic Habitat Toronto, and that process has repeated. At one point a few years ago, we actually tried to back away. We said, you know what? I feel like we've done a lot of this kind of work, you know, like, are you sure, you know, like, you know, do you want to keep throwing money at us? And they're like, yes, we need more. Here's what we need. Here's the next set. And we're like, okay, sure, that's fine. We just didn't want to be too greedy. And it's fascinating listening to the practitioners tell us about how our science is used. This is sort of where Aquatic Habitat Toronto fits in, where it brings together the generators and users with a focus on implementing the Toronto something, I forget what that is. Anyways, it's the guiding document around doing restoration in Toronto Harbor. That involves consultation and the actual knowledge co-production, and then leading to effective ecological restoration. And the consultation side, here's just some ways in which that is done. So projects are discussed at monthly meetings, identify restoration goals, there's feedback, proponent refines concept and design, and so on. So this can extend past research and involve folks that are doing work. So consulting firms that have been hired or government agencies. So it doesn't have to be science necessarily. And then here's where knowledge co-production fits in. So we collectively identify those gaps, identify knowledge, the goals, locate funding, do the work, often together. We're usually working aboard a TRCA research vessel, working hand in hand with other government agencies. We analyze the work, the results are presented and shared, discussion, and provides opportunity for folks to bring this knowledge to bear on their day-to-day decision-making practices. Just showing you that Toronto Harbor has changed, and on the right is an example of all of the habitat. In many cases, is creation because restoration is somewhat challenging given that the current configuration of things. So it's probably more appropriately characterized as habitat creation or rehabilitation. We use telemetry to do this work, and here's just an example. So in 2015. We did a study where we looked at depth use of overwintering largemouth bass. That whole process involved constant monthly interaction with the team. Depth use was incorporated into habitat design in 2016 using the information that came out in 2015. Andrew Roos did some work where he evaluated the efficacy before and after of some restoration, and that led to the identifying need for carp barriers. There were a lot of carp going into some of these restored sites, making them less desirable for native species. Did some before-after work. That led to some additional work to open the lake, and then some full evaluation of before or after. So the work certainly continues there. So some of the benefits, advance notice of projects. So even if it was co-production being done by a smaller subset of that group, others had the opportunity to become involved. Lots of expertise, and really a lot of that bi-directional knowledge exchange. There were some challenges. The biggest with government is that every other week, the representative from a given organization tended to switch. So consistency and agency participation was challenging. Getting folks to commit to coming on a monthly basis, changing political landscape, stability of funding, and differences in goals and priorities across agencies. But I think it's been quite successful. I know it's certainly enriching for all of the trainees that are involved, getting to interact with that many end users on a routine basis. I told you I was not going to dig into any Indigenous co-production cases because I don't feel I've done any of them well enough to justify sharing them. But I do want to touch upon a good case study that you could check out. So that's Jacqueline Chapman on the right, and you can see her in Joe Haven in the Arctic, sitting at a table with some Inuit community members. And in this paper, she talks about operationalizing that knowledge coevolution framework that I shared with you earlier, the earlier Chapman and Schott paper. And in this paper, they reflect on things that they changed. There were changes to their overall research objectives based on their interactions with communities and the local natural resource management boards, including even the focal fish species that were being tracked and studied as part of this. That changed as based on community interests and desire. Concerns where contaminants in food were brought up, which stemmed started a new direction in research. And there was a lot of capacity building and, in fact, interest in using a local facilitator. Oftentimes when we think about facilitation, we bring in some third-party external individual, but they worked to build capacity in the communities and began to make use of local facilitators. So some interesting things that they did there. To start wrapping this up. It is very possible to recognize co-producers. I pointed several examples out where that's been done in papers we've done. We've published with Hedrick from Muskies Canada and all sorts of fisheries managers. We recently mused about the importance of being more inclusive with authorship and valuing different contributions and contributors, including knowledge brokers, the time taken to do co-production work, and those that engage in it, including community members or practitioners, and of course, those that actually do the relationship building work. The work can't proceed without that. That's as important as doing an assay or collecting a fish in a net, as far as I'm concerned, or putting pen on the paper. Just want to show you here. This is something. This was another COVID project with folks at Carleton, and we basically took the word conservation. We broke it down by letters and created an acrostic. And you can see there that it was helpful that conservation starts with the letter C. But I really love that it does, because it really sets up this is what conservation means to us, and putting co-production at the top, I think really. All of these things are important, but putting co-production at the top, I think, really emphasized, for us, how important that is, and we talk about that in that paper. So, last slide. Even when co-production is done right, you're not always going to bridge that action gap in the way you might hope or expect. There will be, partial success is better than complete failure, but you got to try and be willing to learn. And this is a quote from a news article in that Nature magazine piece that I referenced earlier, and I really like this. The end product of co-production done well is almost in the intangible. So with that, I'm going to figure out how to stop my screen share. Stop sharing. Come back here. Apparently I talked for 59 minutes. And I'm going to go to Paige and say, or Daniel, and say, I don't want to throw you off timing wise, and I know that was a huge brain dump. What's next? Do we have time for a few questions and thoughts?
We can sneak in a few questions.
Okay. Thank you. So
we've had a lively chat too, Steve. So don't you worry. It's
okay.
All
thank you.
Good.
Thank you for doing that, Tina. Lovely. I look forward to having a peek over there in a moment. Any thoughts, perspectives, any stories to share? Anyone? I see a hand. Mark Shrimpton, I think, has his hand up.
You're muted still, Mark.
Thanks very much, Steve. When you started, I had no idea what you meant by co-production. And then you started talking and I go, oh yeah, we're doing that. Oh yeah, we're doing that. And I'm very pleased to see people, and Pages, who everybody knows well, her master's thesis really developed out of ideas that came out of the BC Hydro and the Fish and Wildlife Compensation Program. But I wondered, I mean, we all have to become experts, don't we? So is there still some room for that pure science in your mind?
Absolutely. And I like to think about science as a continuum from fundamental to applied, and different projects will fit along that continuum in different places, and fundamental understanding can be applied, but perhaps on other timescales or time frames, and oftentimes unexpectedly. So this is not and do we need to have if you're doing a lab experiment to understand carbonic anhydrase, do you really need to set up an advisory board with fisheries managers and indigenous community members? Probably not. But when you start to do work on lands, in waters, and try to make those connections back to how systems work, that's where, and address those applied problems, that's where co-production really shines. And remember, all of this stuff, almost all of these concepts with bridging the science action gap, this has come out of the medical sphere, where alarmingly there's a lot of science that's done that does not find its way into practice, even though it could make our lives better. And so much of this is leaning on them. And certainly there's so much important foundational, fundamental work that comes there. So when things like the Naylor report come out that, that jump up and down and say, we need more fundamental science, I jump up and down too and say yes. And when they throw more money in the Alliance program, I also jump up and down and get excited about that. And I think almost everybody on this call works variously along, and you're not just one space. It's not an either or, and those projects also support each other. The fundamental supports the applied. The applied allows us to do big science sometimes that we couldn't do because of the lack of fundamental funding, and at the same time address an applied question. So it's all interconnected.
Thank you. Daniel, you're next. You're muted.
It's interesting how things have changed. And the idea of co-production was not well developed when I started doing research, but applied research was. And having partners who could provide the knowledge gap that I had as a basic scientist. And I'm finding in the last 5 years, I've had practitioners and user groups reach out to me and initiate projects, as you were describing. That is sort of breaking down the so-called Ivy Tower isolationism, and is leading people who have applied issues to think about approaching pure researchers, or at least university researchers, on a not necessarily a co-production framework, but on a partnership framework that is essentially co-production. And it's fascinating for somebody who's been in it for as long as I have to see this sort of sea change, where it's no longer us going out looking for a problem or looking for a partner. What are your thoughts on this?
Yeah, absolutely. My career has been a little shorter, but I've also seen that change as well. And I do think that there's Yeah, I can think back to my PhD. I did some co-production then, didn't know what it was called, but I think, or elements of it. But I think now that there's scholarship around that, it can help to really catapult things forward, to reduce the trips and falls and failures, and provide a structure for engaging in this kind of way and get us to a better place more quickly. Because a bad partnership between an academic and, say, an Indigenous community will have consequences for future relationships with researchers and so on, and that may harm the community, in fact, and there's lots of managers that will tell you stories about how academics are too book smart and and don't know how the real world works. So I think a lot of this is using that scholarship to frame what we're doing and help get us there more quickly and more effectively, because it's just like being in the classroom. There's and none of us were trained as educators, yet there's a wide range, there's much educational theory that underpins everything we do. And I remember a democratic classroom, and I'm not remembering any of the term, yeah, you know what I mean. So there's theory behind that as well, and I think that when one takes a step back and reads that, it sort of gels, but it's not just like putting a label on it. It's actually moving things forward and advancing how we do things so it can be done better.
Okay, we can sneak in one more question if it's quick. Does anybody have anything pertinent? Margaret.
I'm not sure if this is a question so much. As I'm listening to this, I think that research in general is becoming more collegial, that it's not that sort of lone wolf superstar sort of idea of a researcher anymore. And I hope this is sort of coming out of EDI, and that we're just tending to be more collegial and forming partnerships that are mutually beneficial, and this is now expanding beyond academic collaborators, but really recognizing what others can bring to the table, not just to help us with our research, but then that our research can help them, and then really focusing on that collegiality, teamwork, and mutually beneficial relationships. So it's really exciting to see.
Yeah, totally agree. Research and science and problem solving is a team sport. And I think the team we're now realizing can extend across disciplines, and it can extend across institutions, and it can extend from inside the ivory tower to civil society and the decision-making sphere and supporting rights holders and so on. So yeah, cool.
Excellent.
Thank you.
Thank you, Steve. Great presentation. At this point, we're going to start to move to the breakout rooms. We have one for the STP-Chip and one for the eDNA. So if you would be able to return to the email that Paige had sent you, there are separate links for those rooms. The rooms are open. Paige is in the STP room, and virtual Joe is in the eDNA room, while this Joe is guiding you over there. Upon the conclusion of the breakout rooms, we will return back to this main link for the final session and then a wrap up. So please select your roundtables, or you can just wait here if you're not interested in any of them, but there won't be much going on. Thank you very much.
It's Heath again.
I return.
Okay.
But I'm going to take a short break. I'll be right back.
All right.
So you're on your way for a minute there.
Okay.
Dan.
Thanks
for
the
that, Steve. That was really interesting.
Thanks.
I had the same sort of response as Mark, where I didn't realize that I was doing some of those things already.
Yeah. Well, yeah, it's very interesting. I see Paige is there. I think Carolyn might be there too. And Carolyn's project really developed with one of my former undergrad students approaching me about a completely unrelated project, and then Carolyn going out there and chatting with them. And they go, "Hey, we've got this problem," and it's really cool. So, but I think it takes a while before you become recognized and you feel that people will actually come and say, "Hey, we want to work with you." So, take some people a shorter time than others, Steve.
I agree with that too. I sort of thinking that as well. Like when we come up with something, any type of biological question, we answer it. We don't get a result within a year. That's for sure.
Yeah. Yeah. Mm.
Hey, Tina, I just had a look at the chat. Wow, well done. Thank you. Thanks for getting that going.
Yep, you're a techno marvel there, Christina, and handling the chat.
Yeah, there's some great resources added and great perspectives in there.
Oh, thank you. Yeah, sorry, my earphones weren't working. They're getting really low with this kind of like full-on day, so I might have to be switching. All right, I'm sorry to interrupt. We've got a whole bunch of people on the chat already, so I'm going to take a back seat and just enjoy.
We're just waiting for Dr. Heath to return. Okay, so I think our general plan for this was for me to facilitate, moderate the discussion. Daniel has graciously agreed to take notes, and we're hoping that we can, at the end of our hour, we can come back with some sort of a breakdown of some of the main comments that have been discussed during our roundtable. So I guess I was thinking I could just share a really nice graphic that Paige made. And I realize we've had all kinds of technical problems, but
if it doesn't work for you, Ken, I can pull it up.
I was gonna say, Ken, you're being judged right now.
Yeah, well, failing in front of a crowd is what teaching has taught me.
That was great.
You can see the slide that was made?
Yeah.
Awesome.
It's very small. So I hope you don't expect we can read.
No, I recognize that. I'll hopefully try to walk us through. But so one of the tasks for the Activity 3 roundtable was to talk about the current status of chips, and then also the species and stressors that can be examined, and then thirdly, the potential future applications of the STP chip. And so Paige made a really nice breakdown of the different types of chips that have already been produced for the GEN-FISH project, based on available resources and types of projects and need for researchers. Obviously, the salmonids have really dominated the chip production. And so we've been able to produce now chips that are involved in just a general stress response, circadian rhythm, neuroplasticity, osmoregulatory function, and immune function already for the salmonids. And what I really like about this graphic is that it shows that not only have we produced or designed a chip for some of these types of processes, but we're already on to the second and third iteration of some of these chips. So we're at that point where we're seeing what's worked with our initial design, and then we're moving on and trying to optimize it for future iterations of the chip. So hopefully we'll just by the end of the GEN-FISH project, we can land on chips that work every time, and we don't have to worry about, or we don't have to hold our breath every time we order a pack of 10 that some of them are not going to work. We know that these assays are going to work. And then on top of that, we've managed to produce smaller versions. So these are 28 gene chips for perciforms, general stress response, and then one that's a little bit more geared towards thermal stress. An initial one for sopriniforms. Again, this is going to be a general stress one. And then some recent work in collaboration with Nick Bernier in my lab, we've been able to produce one for acipensor sturgeons. And so this one's actually been tested out now, and it seems to work quite well. And I know sort of some newer updates that have come along for these chips. We know that the transport stress chip for salmonids does in fact work quite well. We don't have results necessarily to show you, but it's working quite well. And we've already ordered our next version of a salmonid thermal stress and hypoxia chip, and that'll be arriving soon, and that's going to be able to run a lot of samples and from a lot of people that are on this call right now. And so that's sort of my my overview of the current status of the chips and some of the species that we're able to work on. Oh, and I guess I forgot to mention Alyssa Weinrock did a great job going over a future development of sea lamprey chips as well. So we'll be able to add that to our pool of resources soon. So we already have a hand up. Daniel?
Yeah, and I'm just going to jump in. I was looking at the list of people who are on this Teams breakout room, and some of the people probably don't have a lot of understanding about the nuts and bolts of the chips. And so I was just going to throw in the idea, the original idea behind using the open array chips as opposed to doing RNA sequencing. And so if you don't mind, I'll jump in 2 minutes. I just gave a talk at DFO in Newfoundland where a lot of people are using RNA-seq in fisheries applications. And my argument there was the RNA-seq approach is when you're trying to identify something at a fundamental level, almost basic research. The idea behind producing these chips, for those of you who aren't using them, is to provide a tool that somebody who's not a genomicist could take off the shelf, potentially even from Thermo Fisher, and run it through a facility, either fee for service or a partner, and get data that is amenable to interpreting without having to have extensive bioinformatics skills. So the data will be making sense to people who are used to basic physiological data in a way that an RNA-seq run with 400 million paired reads would be more challenging. And so that's why, in my opinion, this fantastic slide that Paige put together and Ken described is so exciting, because we're developing these tools that others can take and run with. So that's my little bit. I'm going to go back to being the secretary.
Oh, no, thank you for that, Daniel. I sometimes jump ahead right to some of the nitty-gritty details. And yeah, just for everyone's benefit, these were largely designed in conserved regions of the genes for these different species, so that these assays can work across different species, which is not a typical approach when you look at transcriptional profiling studies. So again, it's pretty exciting to be able to use the same technology for various projects that we might have in our lab. My lab, for example, we work on a lot of different species of salmonid. So being able to just use the same resource and start to develop analysis pipelines that will work for all the different studies is going to be really, really helpful. Dennis, your hand is up.
Yeah, just a quick question. This is great. These 4 orders are very different in terms of species diversity. Do we have any data on, is there different specificity across the different orders? I mean, Acamentiformes, you'd expect to be highly conserved. Cypriniformes, not at all. And so Do they work across the whole broad range, or don't we know yet?
Because of some of the challenges, especially with cyprinids, we haven't moved as far along as we would have probably hoped, based on the initial grant proposal for that particular order of species. I would have to defer to Daniel about what species have been tested on the cyprinid chips at this point.
Thanks.
So the short answer was our original intention to have one chip for all freshwater fish species was laughable. Although there are some genes that probably would work, but in order to make them useful in terms of the genes of interest that was generated by us all, we've run into challenges. And as we develop these, I'm pushing people to get cDNA from a real broad range of species and just try them to see if we get any signal. The first perciform chip that was developed, Shaheen did that, had fairly limited breadth, and now Patricia's redesigning it using new data available. So these things are sort of moving targets, and it's a process of potentially doing RNA-seq on some understudied species to make them more broad.
I think that's a great point, because a lot of the initial studies, we were relying pretty heavily on available resources through publicly available repositories. And so then if you're downloading just those ones, you tend to get a lot of hits for sort of the well-studied species. So I know for perciforms, things like yellow perch would be, a species that there'd be a lot of genomic resources for, and less so for some of the suckers, for example. So I think as we sort of do some of the transcriptome sequencing, and we can add those sort of less understudied species into the mix for designing the assays, we might get a little bit better performance across the diverse range of species. I can't actually see whose hand is up.
Trevor is next.
Yeah, no problem. Yeah, no rush. So quick question. So two things. One, are we going to set up a sort of either a GitHub or some kind of central portal to show people how to use this? Because people in Activity 4 may not be able to redesign their chip, if it's not working, but they need to have sort of a centralized website where they can go learn how to do this, so here's the RNA steps, here's all these steps. So that's the first question. The second question is, as we've learned more and more in the last year or so, are there certain tissue types that are more prone to problems than others, so we can avoid them or not? We're very fixated on these non-invasive ones like gills, because some species are endangered or not, or expensive. So can you comment on those two things? Are we going to set up a central portal, and are there some tissues that work better than others? Thank you.
Great questions. I'll take a stab at answering this, Daniel, and you can jump in if I miss anything. The plan. Okay, I'll go with the easier answer first. With the tissue types, largely we're trying our best to pick genes that will be differentially expressed across tissues, so that we have sort of a broader use for the different types of chips that are produced. We do have some challenges. There's going to be certain tissues like brain that are going to have a lot of really specific genes that are associated with the brain that are just not going to work across other tissues themselves. So in those cases, it makes a lot of sense to have a brain specific chip. And then just from our standpoint, what we're learning the hard way is that some tissues are harder to deal with after they've been preserved in something like RNA later versus flash freezing them. And so we're just trying to work our way. It's more about whether we can get a solid RNA extraction out of the tissue versus so much as whether it works on the chip. For your first question about a GitHub or a user portal, so now that we're producing data and we're able to start working on the analysis pipelines, we should be able to get more standardized approaches for analyzing the data, and through a different grant through the Great Lakes Fisheries Commission, we're hoping that. Well, sorry, I'm getting my grants mixed up. Through an alliance grant that we're hoping to submit, the plan would be to actually bring in somebody with computational abilities to be able to start to design things like graphical user interface type web-based tools for doing some of the analyses as well. So hopefully that'll help with standardizing some of the analysis approaches.
Yeah, thanks. That makes sense. I guess my only concern is there's some grad students that are they've got their tissues now, they've done the RNA isolation, but they're kind of stuck at that next stage. And I don't want to speak for other lab groups. I've spoken to 2 other lab groups who have the same issue where they've got the tissue, they got the RNA, but now they're kind of in the holding pattern because they don't have the capability, including myself, to really jump to that next stage without inserting ourselves into Daniel's lab or somebody else's lab. I didn't know if there's a way to rectify that. So maybe a bigger question than what we're having discussion today, but I just thought I'd bring it up that there's a lot of samples at that RNA isolation stage that are kind of stuck. And the informative thing you also just said is we also wanted to know if we should flash freeze or RNA later different kinds of samples. So that's something we can follow up with you. But thanks for the answer. It's very helpful. But I just think we're stuck in a holding pattern for a lot of groups right now.
Right.
And to give a little background, as you know, Trevor, the UWindsor Environmental Genomics Facility is in the process of coming up with a fee-for-service to go either from tissue right through to RNA, cDNA, and then running the chip. For labs that don't have or don't want to invest in the genomics or transcriptomic infrastructure, you could ship your tissue or your RNA, or for that matter, cDNA, and have that done for you. Shelby's hoping to have a chance. She's waiting back for getting quotes for kits. So we're hoping to have that information before Christmas, just to make
I was
things.
just working with her on that, Daniel. Yeah, just like recently. So she's going to have pricing, she said, in the next 2 weeks or so.
Perfect. Perfect. The other thing that I'd like to point out is that our experience, and Ken, you can jump in, and in fact, everybody else in here who doing RNA cDNA conversions, we're finding that we can get by with lower quality RNA for the open array chips than you can for RNA-seq. With one caveat, there are some very low expressing genes that appear to sort of wink on and off, and we haven't tested, but we think it has to do with RNA quality and therefore the quality of the cDNA library that's generated. But generally, if we do RNA later properly, that is, we don't stuff a whole fish in 15 mils of RNA later, we're getting relatively good cDNA for the open array applications. And Paige, I saw your hand up for a minute there.
Yeah, I just thought I could jump in and at least discuss kind of the internal GEN-FISH sharing of what's working and what's not. John and I are kind of in development. I'm going to boot you out for a second, Ken. We have this matrix that you can hopefully kind of see coming up, where preferably students or HQP will be able to go in and select the chip that they ran, and then view the genes on it, their main gene function, and then for whatever species that was assessed, to be able to choose which tissue quote unquote worked or didn't. That's kind of a simplified version of it, but you can go and say for everything here, these things worked, this didn't for that gene and that species, if that makes sense. So this will be on Teams for everyone to be able to use and fill out, hopefully soon, like within the next couple months. And then we'd also like to incorporate the that kind of infographic of the STP chip development, perhaps as an interface for that, eventually to be able to put online, to be able to click on one of those and see the results from it in a more detailed way. So hopefully we're going to have a better kind of communications system for how the chips are going, if that makes sense.
Thanks, Paige. That'll be exciting when that's up. Celine?
Yes, I just wanted to say that it's a great idea. I think that this will be extremely useful because we just share some of the results that we've got, sometimes with only you, Dan, or Ken, but it was never available to everyone. So this will be extremely useful. So as soon as it is ready, then just tell us. In fact, it seems that it's ready. Then you could put it on team right
Certainly.
now.
Nick?
Yeah, I wanted to come back to Trevor's question in terms of like which tissue works best, and also comment on maybe what something that Daniel said in terms of the quality of the RNA. So to come back to Trevor's point is that some of the chips have been designed knowingly that some of the genes would work good on some tissues, but very poorly on other tissues. It was designed like that on purpose. And to give a very specific example, the latest salmonid chip, or at least one of the versions, has a few different osmoregulatory genes on there. You will get data if you, if you have gill tissue, and you won't get data if you have liver tissue, and it's on purpose. It's just that we wanted to have different functional groups on the chip, and that some of the genes will be expressed in some tissues and expressed at very low levels in other tissues, and will simply not give you any data. So it's not because the assays don't work. It's just because the selection of genes is meant to give you data from different functional categories, and that of course different tissues carry out different physiological functions, and so that's important to keep in mind. And the second point is that some functional genes have fairly low levels of expression by nature. That doesn't mean they're not important. It just means they're more specialized in their function. And that's where the quality of the RNA is very important, because just like Daniel said, we've had the same experience in my lab, whereby for those genes, having very good quality RNA, cDNA, will make a difference in terms of the number of samples that will give you valuable data. And my last point before I turn my mic off is that keep in mind when you analyze all the data moving forward from the open arrays, that the arrays are in general very good at detecting upregulations of genes, but not so good in detecting downregulation of genes, and that's just based on the nature of the way these things work and their sensitivity. So it's important to keep that in mind, because if you're looking for downregulation of certain genes, and these genes are expressed at moderate to low abundance, you probably will not get valuable data.
So, Nick, I have a question for you on that. And that is, let's say your control sample is giving you expression, but very low levels. And then you do your challenge, and those genes are now undefined. So they actually didn't, quote unquote, work. Could you not interpret that as some level of downregulation? Or would that be the sort of thing reviewers will pounce on you?
I think you have a valid point to say that there can be some down regulation, just because if you have low CT values for your endogenous controls, that means that your endogenous controls are going to work amazingly. Now, if you have absolutely no values for your challenge that you just did, then you might get some possible solution for it.
But yeah, I think Nick, you bring up two really, really important points when dealing with these chips, is that it's some of the assays that are on the chips already might not work for really subtle changes, and then it's also perhaps not ideal to have really low expressed genes as targets on the chip, because it might not always show up, and then we have to deal with how do we deal with this challenge of how do we analyze data when we have no detections versus detections. And then the other one is just, another part of our problem in the strategy of producing these chips is that we need stuff that's going to have decent levels of expression, and then we have to order these chips at least 10 at a time. So we can't really have too many highly specific chips for a single lab group, unless they're planning on really running a lot of samples.
If I may add, it's a challenge designing these things on so many different levels. Genes that are often highly expressed across all tissues are constituent and often don't have much don't give you much physiological insight. I mean, there are exceptions. Heat shock proteins are expressed in every cell and are like your perfect genes for the chips. but there's a large number of genes that are highly important physiologically that have relatively low levels of expression, and in the end, like everything else, it's reaching that happy balance of a suite of genes that are going to be informative, but at the same time will give you robust data, and it takes time, trial and error to find out what that magic balance of target genes are.
And hopefully over the next year we'll be getting a lot more complete data sets that'll help with informing which genes are informative for responding to certain types of stressors.
And the suite of genes that's on the GEN-FISH list was not simply made up by Ken. At least I hope it wasn't. Because
no.
The plan behind it was to go to each of the labs of people who are doing transcriptomics, who have expertise in groups of genes, and saying, what genes have you had success with in terms of, doing single qPCR assays, whether it be CyberGreen or TACMan, and some of the genes were thrown on just because we knew they were important. We did not have a lot of experience with, but a lot of them we did. And I am actually amazed at how well it's working. And there are, on every chip of 28 or 56, you're going to have failures depending upon your species, your challenge, your tissue, your RNA quality. All of those are going to add up against you. But as long as you don't go in there saying, I've got to get this gene, if your intent is to say, I have to know, what sodium potassium ATPase 1A is doing, then that's when you do a single assay. But if what you're doing is saying, what's the gill doing when I raise Chinook salmon in fresh water for their whole life? Some of the genes on there may not be informative, even though we can get data for it. But all of that's going to add up. And as Céline said about Paige's and John's spreadsheet, that's almost going to be the deliverable for activity 4. at the end of the day, people may not use our chips, but they can go to that spreadsheet and say, okay, these, I'm going to pick these and I'm going to print my own chip.
Yeah, and I think that's going to be a super exciting deliverable for this work, just being able to custom design chips in the future based off assays that we've designed. I think that's going to be really, really exciting.
But Mark.
Well, if you wanted to continue and say something else, Daniel, that's fine. I want to ask a very specific question of Ken, because you introduced this idea of your general stress chip versus the thermal and hypoxia stress chip. Are you looking just at less genes, or have you got more thermal and hypoxia responsive genes on that chip?
It sort of the general stress one was trying to target a lot of genes that are representative of various types of physiological processes. So for our thermal hypoxia chip, it's bumping out a few of those genes that we weren't sure would be as useful for us, and trying to focus more on sort of the high responder heat shock protein type genes, and then things like more specific to aerobic metabolism versus anaerobic metabolism and those types of processes. So it was just a matter of taking what had been done initially by Shahin was to produce a 112 gene chip, and then we can figure out what assays were working for salmonids, and we have I'll briefly talk about it tomorrow during the activity 3 presentation, but essentially he tested it across 4 different genera of salmonid, and I know I'm using my taxonomies wrong. I can feel Nick responding to that. But we tested across 4 different genera, and just to see how well it worked. I think there's 8 species, 4 genera, and we tested the 112 genes, and that sort of created our initial database of assays that we could use. And then from that, we sort of tried to target sort of the assays that are more specific to thermal stress and hypoxia.
And to throw a bit more, because we've done a couple of those more specific stress. That's also logistic. So we go to fewer genes, we could do more fish more quickly. When we know what we know we're hitting them with hypoxia and thermal stress, then some of the other genes, detoxification, that are going in the general, we don't really need it. So I think the general stress has everything in there, because perhaps you don't know what all the stressors are.
Yeah, well, and that's kind of my point in asking is we have a lake where, we know hypoxia is a problem, we know temperature is a problem, but there's a whole bunch of other things going on. And just to keep, I think Brian Dixon's on here, they may be immunocompromised as well. So, is it better to go after a larger suite of genes and less samples per chip? Yeah, but where my questions are coming out of, Ken, because we've been emailing back and forth. So
right,
it's what to do with that valuable cDNA.
Absolutely, and I mean, I think at least from a initial screen, if we just use the available thermal hypoxia chips, you can make sure that you're going to see a difference between your groups of species, and if you think that there's going to be other problems associated to it, then we can maybe figure out ways to address either use the immune response chip that has been developed already on those samples as well, or you didn't say pollution, but I'm always excited about polluted fish. So we could also look at detoxification genes.
We're in the middle of BC. It's all pristine up here.
Yeah, except for pulp and paper, which I hear changes the sex of the fish. Details. But the other thing that I say this all the time, I'm going to say this again, is remember that GEN-FISH has a goal of trying to figure out how these things are working. And consequently, failures are not failures. So, you know, when Céline was able to work with a striped bass and it didn't work out quite the way she wanted, that was really valuable because it told us that our Perciform chip just was not doing what it needed to do. And so the other side of the story is we have 18 months left. We've still got, Joe, correct me if I'm wrong, but somewhere in the neighborhood of about $300,000 worth of chips. And so printing a chip and trying it is, even if you come away and say, okay, we did this and nothing worked, it's a waste of a little bit of time, but it's better than waiting, because even negative outcomes are going to be really valuable for the final point of the project.
Phil?
Hi. Yeah, sorry, hi. I had a question actually about low expression genes. Like, so that came up, I think Nick mentioned it, genes that, you know, you can't detect always on the chip. And in particular, I'm thinking of, we've tried to detect genes like cytokines, and we find it very hard to detect them reliably, probably because you need to have a certain level of inflammation before they're expressed at high enough levels to be detected. So I was just curious what people's thoughts are about things like that. Just to get back to the issue that, you know, you have a finite number of genes that you can test for in a precious sample. And, you know, I guess it's very important to have a set of control genes so you know that the sample integrity is still good. And then you're trying to interpret, you know, holes because you have, let's say, you know, 100 different samples and you're detecting cytokines in only 20% of them. Is that data useful? Is it, you know, can you, what do people think about that? Is that if the controls are working, if that, you know, panel of like general genes are present, is this still interpretable data, or is this like dropout effect a reason to just, you know, not use any of it really?
Brian, I know you're on the call. Is it okay to defer to you on this one?
Sure. Well, I was going to say, I mean, I've run into this problem before. Like, I had a student working with interleukin-6 just in regular qPCR, and she could never get any signal at all, like within 40 cycles for her controls, because there just wasn't any gene expression there. But she'd get expression in the stimulated ones. So I think it is a problem. The genes I picked, I think, shouldn't really be that bad, but you're still going to have the problem. But I think the thing is, I mean, I just kind of went with, say, just noting that there was an increase in expression because you see something when there's nothing in the controls, but it's very hard to quantify, right? I mean, but it is useful to know it's expressed in certain places. I mean, I've been making protein assays for these, and for interleukin-1 beta, the basal level is 6 picograms per ml in blood, so they're just low levels resting in terms of genes and proteins. I don't know mathematically how you would get around it, really. I mean, you know, other than doing 45 cycles or something, but that's ridiculous as well. So yeah, I think on the chip, you run into the same problem. You're going to get certain tissues or controls where there's nothing there at all.
Yeah, my feeling was like, if as long as the control gene is expressed at a, I guess, comparable level, then you can't use the data basically because your control is okay.
Yeah, no, I mean, and you know, the chip's working. You're getting other genes there. It's just that particular gene is problematic.
Cool. Thanks.
Celine.
This would be perhaps something interesting to add on the chart we will produce for at the end of GEN-FISH. But in a lot of the, for example, for the two brain chips, the two sets that we used for two different projects, we do have, in one case, the RNA-seq data, and for the other one, we have the qPCR data. And for the one we will do with Ken and Brookchar, we do have the qPCR assays for many of those genes. Then we do have real, you know, it's the same experiments, the same animals. It's the same RNA that is used. So we do have very clear comparisons, and it will be easy to get some responses on, and trying to find some responses, for example, for, okay, what do we do if this works or not? What does it mean? Because we do have exactly the same animals in the same conditions, with two very different methods. And we will have this for many different projects. And it's really a very strong validation that will help us, to really define how we can use the results, for example, and answer some questions that we do have. But we will have these kinds of data.
Yeah, that's a really great point, and those would be really strong for kind of a final report for this project, just to show that it's representative of a whole transcriptome response. Thank you, Celine. Tina?
Celine's comment really segues nicely into mine, in that we have a lot of academics right now in our workshop. We're not hearing from our end users. And so at the end of the day, we're talking about validation, but then we have to translate this into what our end user and the type of information our end users need. And so do they need that really? We have the validation, we need to be able to validate it to that micro level, but we have to think about then what do we translate it to in terms of, is this an effect, and how much of it, and should we be worried? If this is being meant as a prognostic tool, that they should be worried about something coming down the line, as well as a diagnostic tool. It depends on the intent at which the STP chip is going to be used. And so just to keep in mind for all of us that at the end of the day, we have to translate our validations and our findings into something that's actionable for our user groups. And will that mean a twofold change, a threefold change, either upregulation or downregulation? What are our limits to say that this hard, like very, very little expression in the cytokine, is that meaningful, yes or no? And just to keep that in mind when we're all going to be discussing more collectively in terms of what the chips are actually telling us, and is it useful for our end users?
Yeah, and to that end, I'll add, for some of the immune genes for salmonids, like for example, I do have an ELISA, like I said, for the protein for interleukin beta and a couple of others. So we can even go a step further. If you guys want to, I can teach you, or you can send the samples to me and we can do it. You can do like the chip, the messenger RNA, and the protein, and the messenger RNA doesn't always correlate with the protein either. So I think you could even take some of that stuff a step further.
That's all really great points.
We're starting to run out of time. What I'd like to do is ask people to pop up with if they have any particular questions, suggestions, or concerns about how activity 3 and 4 are going to play out over the next 18 months.
It's only 18 months.
We do have some water under the bridge.
Yeah. So in my experience, again, it's like it's easier to generate the data. It's getting easier and easier to generate the data and run the chips. I think we've overcome that those hurdles. The biggest hurdle in the next 18 months will be to analyze all that data. So when Carol Bess was in your lab, Ken, right, she analyzed a large data set, one of the first ones produced by the arrays, and came up with, using R, ways to streamline the analysis in R. That's knowledge has to be shared so that the next step can be streamlined. I think we're all going to hit a wall with a whole bunch of open array data in the next 12 months, and really hit a wall in terms of converting these massive Excel spreadsheet, into something that we can interpret as scientists. So I'm just putting that out there.
And that's a really great point. And I think especially because as Carol is so diligent and so meticulous with how she prepares all of her files and everything, I think it'll be really useful to at the very least publish her code with the future papers that are going to come out, and so that it will be available to everyone and it will be accessible. And she's done a really great job of automating a lot of the format restructuring that's required to use things like the program for looking at assay efficiencies and those types of parts of the analysis. So I think we need to publish the code. That's just my opinion. I'm going off script here. Sorry, Daniel. And then that way it's available for future researchers, and then have that available through the team site as well would be really useful.
I think, Nick, it may go beyond simply dealing with the data. One of the things we need to do is we need to get away from delta CT thinking, because as soon as you see not determined, you figure it failed. But I think what's coming out of this discussion is it isn't failed. It just means it's so low it's not detectable, which is essentially a zero, unless the assay failed. But we have to not only be able to analyze the data, but we need to put it in a context of saying, what does it mean if interleukin-8 is being expressed at this level? And that's where we come back to John and Paige's spreadsheet. And somebody sometimes going to have to analyze all the data we input from all our chips and look for patterns, and to look for patterns of genes responding in predictable ways. Céline?
Yes, this means that we will need a place where we could send all the data, that GEN-FISH has, having a kind of data depository or something like that, that we can put all our chip data, all together, that otherwise it will be very hard to do what you just said. But we need a place. I don't know if it's possible if GEN-FISH has the capacity to do that, but this would be the only way.
I totally agree, Celine. John LeBlanc is on the call. John, can you speak to that? You've done a lot of work organizing everything.
Yeah, so we do have all the data, the raw data that is backed up on Teams. And as soon as we get maybe like a workflow to process the data, we could add that to Teams as well. Yeah, we've been using Teams as our kind of data repository. But yeah, we have all the raw data for every run at UWindsor. So Ken, I might work with you to get your runs that are run at UManitoba as well.
Sounds good.
But I mean, if we want to take taking some information about the whole thing, we will need more than raw data. You will need to know what they are linked to, and if there are some control groups and everything. So all this information is on our side and not on the GEN-FISH side.
Yeah, I was going to say that too. Like my students writing a thesis in a paper, that should be there as well, because my students, as you said, Celine, comparing the chip to qPCR and in several tissues, I think all of that stuff needs to be there. And I think, I'm hoping there's going to be more widespread use of the chips, because so far I'm the only one I think who's used the immune chip. But if other people do, I think that sheet Paige showed us is going to be very important. And I'm going to have to sit down, for example, and stare at all of the summary data for the immune genes and go, okay, does this make sense? And Nick's going to have to look at the stress ones, or the neuro whatever. Like we're all going to have to go in and look and say, does this make sense to us? What patterns can we see across the whole? I think that'll become important. So that will need, like Dan said, we need 12 months of gathering the data, but we'll need a few months for us all to sit down and ponder that as well. Yeah.
So that's an important thing to remember as we're winding down for our end of GEN-FISH. Daniel.
Yes, I think that this will be great. If we do have such information at the end of GEN-FISH, then it will be really great. We will have really accomplished something.
Yep. So a couple of things that I'd like to mention. I'm sort of writing down all this stuff, and something that pops out of me is there's a whole pile of questions that have come up that I want to talk about more. What do you all think about having a sort of informal but regular forum for activity 3 and 4 PIs and HQP, just to get together on Teams, maybe with a theme each time, such as tissue specificity? You know, brain versus liver is a no-brainer. I did that on purpose. But I think other questions come up, and things such as how to preserve RNA in the field. Sometimes you just can't do flash freezing.
Yeah.
So what's the general feeling about having, you know, monthly or maybe bi-monthly informal get-togethers?
Yeah. Be good.
Great. This would be great.
Especially given that we've got money for chips and not much time. Good. Okay. I think at this point we have to shut her down and go into the other room for barriers. And in fact, some of this might come up under barriers for adoption.
Ciao.
That sounds great. Thank you very much, everyone.
Take care.
Appreciate your
Thank you.
input.
Thank you.
Danielle.
Guess we should wait a couple minutes for folks to filter in.
Yeah, and then I'll let you guys take care of the moderating of the room if you don't mind.
Yeah.
Good. Out of curiosity, did we get a sense from registration in terms of indications of numbers to expect for folks showing up?
I think if I'm remembering correctly, there were maybe 9 that were registered, and it looks like we have almost, yeah, that we have maybe that now. And then I suspect there may be a few others. Yeah, we can wait to see if there's a few more, but I will say, as we already have some people here, that we really are trying to encourage discussion during this roundtable. I think the numbers are low enough that we can have a good discussion. So you've all sat patiently and listened for most of the day, and so this is a chance to really participate and ask questions and talk. So please don't be shy.
Do we have a sense of when are we going to just keep the 15-minute delay out of curiosity in terms of when the next session is going to start, or should we be targeting to get back on track in terms of timing for the conclusion of the roundtable?
My feeling would be we'll sort of see how it goes, that if things are sort of coming to a natural end according to the regular schedule, like after 50 minutes rather than an hour, maybe that can get us back on schedule. Yeah. But I certainly wouldn't want to cut off a discussion if we wanted to use the full hour. That's sort of my feeling. And that depends, of course, what the other roundtable group does as well. If the final session starts at 3:30 or 2:30 or whatever time zone you're in, yeah.
Yeah. And I've got a chat going with Paige off to the side here, so there are 27 in the other room right now.
Yeah, yeah. You can definitely give us an idea if we should be when we should be wrapping things up if the other group is wrapped up.
All right, I guess we might as well start now. We've got 10 folks in the chat, so I think it probably makes sense to get things rolling. So yeah, I'll just introduce myself. I'm Dr. Matthew Yates. I'm working here at University of Windsor with Dr. Heath. I'm a postdoctoral researcher. I'm going to be co-moderating the roundtable today with Dr. Margaret Docker, who I'm sure everybody here probably knows very well as a prominent GEN-FISH researcher. So for the roundtable today, I guess we kind of, as Dr. Docker was saying, we kind of plan to just kind of keep discussion fairly free flowing and open. You know, I think we're all kind of just interested in having a discussion about issues and challenges that can come up with the application of eDNA assays in natural ecosystems. And we thought that we would sort of discuss or divide the discussion up into kind of two sections. Again, this isn't hard or fast or anything. If there's solid discussion going on the first topic, then we'll probably just keep going with that. But we figured that we'd spend kind of the first half talking about assay design and validation, so the sort of like in silico, in vitro side of things, in the work that's typically done in the laboratory, and then devote sort of the second half of the roundtable to discussing issues associated with applying eDNA assays and interpreting the data you generate from them in natural ecosystems themselves. And obviously there's a fair amount of overlap between those two topics as well. So we thought we would start by first going over or getting Brooklyn Litkey, a GEN-FISH master's student who has a ton of experience doing this sort of thing, give a short presentation to sort of describe her typical workflow for designing and sort of validating these assays, just to get a handle on the sort of processes involved and the type of work involved in doing this. So I guess I'll hand it over to Brooklyn to start the session.
Yeah, and I'll just add that, that it's definitely an overview, that there's a lot more details involved, but we're trying to sort of, for those of you who want to know more, now know who to ask, and those of you who just want to use the assay, this at least gives you an idea of what goes into developing it, so you know how much you can trust it, that you have a sense for the different steps that are involved in developing and validating. So the idea is to provide enough of an overview so depending on whatever your interests are, you can appreciate it. Yeah, so go ahead, Brooklyn.
Yeah, thanks. And now I'm kind of terrified to share my screen based off of everyone's failures today. Okay, let's give this a go. And now from beginning, how's it looking?
Looks great.
Wow. Okay, sounds good. So taking a look at the participants here, I think that this might be a very basic overview for some of you, but kind of our intentions with this was to provide a summary to those who might be less familiar with eDNA assay design, what the basic workflow of designing an eDNA assay looks like. So when we've had some people approach us to incorporate eDNA in their projects that are unrelated to eDNA, say they want to they're doing some sort of other field sampling and they want to throw in some eDNA in there. What I've kind of noticed is this is what the workflow that people expect eDNA incorporation kind of is. They determine a target species, they determine a target region, and that's geographically. And then either they're going to design an assay from scratch, adapt an assay from a publication, or they're going to use an existing assay. And in reality, it looks a lot more like this. So you decide to use eDNA. A lot of stuff happens, and eventually you can then detect the target with eDNA. And when I first started with eDNA, I definitely felt that middle section a lot more, and now I kind of live in flow with that. And hopefully I don't make any of you feel that middle section throughout this presentation, but if I do, that's why those of us who do this for a living exist, and we're happy to help you through that process. But anyway, for today's overview, I've broke down the process or workflow of designing eDNA assays into 3 key components. First, you define your objectives. Then you're going to identify conserved regions within the targeted gene region that you're going to target with your assay. And then you will then design your assay. So I've formatted the defining of the objectives into that who, what, where, how, why question format. So who is your target species or multiple species? What is your target region? And I'm talking about what is your target region within the genome. So what gene are you targeting? What is it, CO1? Is it site B, 12S, so on and so forth? Where are you targeting this species, and geographically, where are you targeting them? But also, is it in a lab setting? Is it in the field? How are you going to target this species? What type of assay are you designing? Is it a cybergreen assay? Is it a TACMAN assay, or a different type of assay? How are you also going to use this assay? Are you going to be incorporating it on an open array chip? What type of analysis are you going to do? Is it qPCR, droplet digital PCR, endpoint PCR? Are you planning on multiplexing the assay? And why are you designing this assay? Now that sounds maybe a little bit pretentious, but is there a published assay that exists? Is that assay not sufficient? Can you use that published assay to as a starting point? But these are all questions that you kind of want to look at before you start the assay designing process. At this point, once you've answered those questions, you'll need to identify the conserved regions of that target gene within the genome that you're going to be designing the assay for. And this requires having sequences of the target gene region from the target or targets that you're going to be designing the assay for. And you'll need multiple sequences from that target region to make sure that you have a good representation of that gene for your targets, because there can be different haplotypes and there are variations of the gene between populations, and you want to make sure that the assay that you're designing is representative of those differences within the species. But you'll also need to have multiple sequences for non-targets like congeners, confamilials, and other coexisting species to make sure that you're not designing an assay that will amplify non-target species. And that's a key component to validation, but it does start in the assay design process. And once you compile these sequences, you can form them into alignments, either of families, of the genus, of your species, and of the regions. So that includes your coexisting species, so that you can start to see where there might be unique conserved regions of your target species that might be good regions to design the assay for. So here at GEN-FISH, we have compiled some lovely GEN-FISH alignments for certain commonly targeted genes like CO1 and site B that I have the benefit of using, which kind of streamlines the process a little bit. So I can also pull my sequences that I'm looking for from that repertoire of sequences, but also reference those when I'm looking at validating my sequence. So it includes all sequences of the target gene. So let's say CO1 for example, for all freshwater fishes and some invasive fishes in Canada. So that makes it a little bit easier. Then when it comes to designing the assay, some things that you're going to face in this step will be choosing a design software for the assay, and making note of the version of the software that you're using is going to be important, not only for your reference, but if you were to publish this assay or share it with someone else, it'll be important for them to know what version you use, because there can be some differences that can arise from using different versions of the software. But you'll also have to define what makes a good assay. What parameters are you going to follow to define what a good assay is? And you can find what that is from various publications, but it can also be dependent on the type of assay that you're designing, what types of machines you're using. So if you're using qPCR, droplet digital, if you're planning on doing an open array chip, in what protocol you're following as well. And it can also be dependent on what kind of software you're using to design your assay. And as you can expect, it's also beneficial to design multiple candidate assays, especially when you move into the in vitro phase where you're testing the assay in the lab. Sometimes things happen and assays don't react the way that you expect them to, so it's always good to have a backup. But before you order those assays, the last step in designing the assay is always to check the candidate assays that you've designed against multiple alignments. So that GEN-FISH alignment that I mentioned, where you're checking it against a large repository of sequences of different fishes, but also another sequence database like BLAST to check against non-fish species is always great too, to make sure that you're not going to be getting that cross-amplification against non-target species. So that's a very basic overview of designing assays, just to kind of give you an idea of some of those steps that come into play when it comes to designing those assays. Yeah.
Thank you very much, Brooklyn. That was a great little primer on assay design and validation.
Primer. That's a good pun there, Matt.
That was unintentional. I should have claimed that. That was a good one. All right. Yeah, so I guess right now, why don't we open the floor to questions and discussions? So I think the first thing we could do is just see is there any issue folks in the audience here or in the group want to ask about? I mean, we've got some pretty good experts here on assay design and validation who've been doing the bulk of the work on the qPCR side of things in GEN-FISH. So you know, these are great folks to ask these sorts of questions of. So yeah, I'll open the floor to anybody who's interested.
Yeah, and if people are hesitant to start to speak up, I just want to get it maybe even just by a show of hands. So how many people here have been designing their own assays? Okay, yeah, that's good to know. So that most of you, it sounds like you're in the situation where you're using existing assays and hoping for the best, or that you may be making decisions based on the outcome of an eDNA assay. So then, yeah, that what you want to know is how rigorously those assays have been. Yeah, how rigorous the development stage and the validation stage is before you apply them yourselves or use the results in making management decisions.
I mean, I'm super guilty of that. Well, I got a phone call going here. Sorry. I'm one of those classic researchers. I work with salmonids, so everything has already been very well validated and developed far before me. So you know, I use assays developed by better geneticists than I. I guess what's a good thing to do to sort of check to make sure that the assays you're using are sort of well validated in I guess the system that you want to use it in.
Yeah, and if I'll just jump in first with that, and then let others jump. I think one of the most important things, and you don't need to apologize that you haven't developed assays yourself. I mean, that's one of the reasons. One of our big goals in GEN-FISH is to design and validate 2 assays for every freshwater fish of Canada, because it is quite an ordeal to develop these, and that there won't be the eDNA uptake if those assays aren't already out there. My feeling is probably the single biggest thing is transparency in reporting. That, you know, when you have an assay that's published and people say it's species specific, universal species specificity is almost impossible unless you have a taxon that's very distantly related from anything else. There's always going to be some cross-reactivity with other species. So if somebody says it's species specific, I want to know tested against what. So it might be species specific in Manitoba, but it cross-reacts with con-specifics in Ontario or something like that. And so I would want to see a list of sequences that it's been aligned against, or a list of sequences that it's been tested on tissue-derived DNA from. And so I think that this just sort of trust me, it's species specific isn't good enough. There has to be all of those details.
So I've got actually
Yeah.
Anne Frechaude has her
Yeah.
hand up, so I'll let her ask a question. I have a follow-up question to that too, though. So go ahead, Anne.
No, maybe you can go ahead. It was more a comment, and I don't know where to start actually. So maybe if you want to jump on Margaret's comment, please go ahead.
Okay, so just to follow that up, I guess. Oh, no worries, Dr. Love. See ya.
Yeah.
So if you've got an assay and there's a list of species that it has been validated against, are there kind of general rules where, like, okay, you say you have an assay, you have a target species, say you're investigating it in an invasive area where it doesn't naturally occur, so it wasn't designed with, the conspecifics in that region in mind. Is there a general rule for how differentiated the sequences need to be in order to have an idea of, I mean, you should always be validating these things with like tissue samples and stuff like that, but as a starting point to be like, okay, would this be a good assay to start working with or try? Is there, certain patterns or in terms of like base pair, how many base pair differences there are in sequences that can give an indication for whether or not an assay has potential in that region, given the set of like conspecifics? I'm thinking about brook trout at West. I work with brook trout at West all the time, but a lot of the assays, that were designed for brook trout weren't necessarily designed with like, I don't know, like bull trout and Arctic char and stuff in mind or something like that, like very closely related species. So so this is something I'm interested in anyways.
Yeah. Do you want to take that first, Brooklyn? Then I can add something.
Yeah. So some papers that have been published, first that come to mind is like the Climus 2020 paper. They suggest at least 2 base pair mismatches in at least 2 of the elements of the assay. So at least 2 in either the forward and reverse and the probe. But I found that's not sufficient. So I'll have assays that have 6 total base pair mismatches, and that's not enough. So it also depends on where the mismatches are. And if you have a forward primer that is sequenced 5' to 3', having those mismatches at the 5' end is going to be a lot more significant than having the mismatches at the 3' end, because the majority of it has already been sequenced by the time you're getting to that 3' end. So the location of the mismatch and also how many mismatches there are is pretty, it plays a role in how likely it is for it to be effective as a mismatch. But I would say if you have like 3+ mismatches in each of, and this is kind of an arbitrary number, it hasn't been published or anything, but from my point of view, if I have at least 3 mismatches in each element, so the forward primer, the reverse primer, and the probe, usually it works well. And you want it at as close to the end where it's going to be sequenced from. So 5' end for the forward primer, 5' end for the reverse, and 3' end from the reverse primer. So usually in that case, it works out well. And of course, the reverse primer, if there are not enough mismatches there, it doesn't matter for the forward primer and the probe, or if there are no mismatches in the forward primer, but there are a ton in the reverse, the forward primer is still gonna get amplified, like because the nature of the assay, there's the forward primer, then the probe, and then the reverse primer. They don't really interact the same way. The reverse primer just amplifies the sequence of DNA, so it's more likely that it'll be amplified with a different forward primer. But anyway, I'm sure there's probably a paper out there that I haven't seen yet that is more has a more concrete number, but really the more mismatches that you have, the better you are, or the greater likelihood you're going to have that it's not going to cross amplify. But really, when you get into the lab, that's where that spiral happens, because even in the best cases, weird things happen.
Yeah, and I think this also ties into, increasing the transparency of reporting, because the more that this is reported, then the easier it'll be to see those patterns. Because like Rena is publishing her master's work where she developed and tested 20 Site B assays for Atlantic fishes. And, and this is one of the nice things with publications now that allow an almost endless number of electronic supplemental files. So, for every of the 20 assays, she's got a table showing how many mismatches there are for all of the confamilial species. And then she does in vitro testing of it. And so a pattern emerges quite quickly that if you've only got 1 or 2 total mismatches across your 2 primers in your probe, you're probably going to get cross amplification. Once you're getting up to 7 to 10 to 12 mismatches, it's good. And then the ones that are maybe in the middle zone, where it's going to depend not just on the number of mismatches, but the nature of the mismatches as well. And then those are the ones that you can really focus your efforts for the in vitro testing on, because in good design and in silico testing doesn't get rid of in vitro testing, but it can sure narrow the number of species that you have to test, because obviously you can't test every assay against all 212 freshwater fishes of Canada. That the really good in silico testing lets you identify the ones that you really need to focus on in vitro.
Thanks for answering that question. And for show, do you want to follow up with your comment?
Oh, yeah, maybe I can try. So maybe first I have to introduce myself, because you may not know me. So I am actually sitting as a position of a genomic specialist in Parks Canada. So I recently joined Parks Canada, and before that I was working in academia for almost 20 years now. And I've been working for almost a decade with Louis Bernatchez at Laval University. So I've never conducted myself an eDNA study or work, but obviously I've heard about that just in Louis' lab a lot. Here in Parks Canada, so I'm doing a lot of things about eDNA, but I'm sitting in national office. So I'm here to give some scientific support into field units with people that are working directly into the parks. And we are classically trying to make a guidance document to implement eDNA as a measure in the monitoring program, into the regular monitoring programs that they are applying in parks. So for that, we could not, this is a big task for Parks Canada, and we could not handle everything in that. So we try to focus on freshwater species. So we're making a guidance document for implementing eDNA measures. It could be for invasive species, for species at risk, or for community approaches as well. So it's both qPCR and metabarcoding, and even metagenomics. We have some parks that are already applying those methods. But if I would just try to focus on the qPCR assay, because that's why we are talking. And so, as I say, I've never done a qPCR assay myself, but so I'm super happy to be here today to discuss all of that with you. And what I've done actually in this document already, so we are trying to describe all the steps that are needed for people to run those approaches in the parks. But maybe I should go in another point before. I should mention that in Parks Canada, we do not have any genetic lab. So it means that every researcher, every scientist in the parks that want to start a project has to make a collaboration with a partner. It could be a government, another governmental partners. It could be academic. It could be any institution that could help. But we need help, and we need to have good collaborators. So people, they are just, by now, they are just trying to reach me, people that want to start a new project because they are still running project already, but people that want to start a new project, they are just contacting me to have a list of good collaborators, depending on the location on the country, because we are just working across the country. So it could be West laboratories for West people in BC, but it could be also happening in the Atlantic. So, and about the qPCR assay, have been asked actually about which lab is actually relevant for doing a good PCR, a good qPCR assay, and what should we consider, and is it relevant to go with this one or with this one, and what is, so I don't have any bias, or obviously I do have bias because I'm coming from Laval University, but anyway, this is not my job, and so this is a point, and there is another point, is that the guy I am working on this, who is actually directly involved into the monitoring program at the national office, wanted me to make a list of all the qPCR assay that have been developed, defined, and even eventually validated for the freshwater fish species in Canada. And at the beginning, I was really, I was not convinced by this idea because for me, coming from WizLab, I've always heard that if you want to make a qPCR assay, you have to make it for especially the special place where you want to conduct, considering maybe the co-related species that could be in this location into the lake, and is there any hybridization happening into related species or not, or considering even the environmental condition or whatever. So I say to this guy, okay, I can make this list if you want based on the literature. And I contacted Peg actually to before making that, just to making sure that you were not doing also this. But for me, it would not work. It would not be worth to do that. And it won't be useful because I can't provide this list to people and say, oh, okay, you want to run a qPCR assay in brook trout in the West, then you can use the one that has been developed in the Atlantic, maybe with not even at all the same related species are, you see. But I made this list. So if somehow you are interested by having this list, I've done an Excel list with every qPCR assay that I've found, but it was before the summer. So I'm just guessing that this list will get updating always. So I just wanted to point that, and I don't know where I'm going with that, but just so you know.
Yeah,
just some feedback for that. I think that's a really great idea to compile those types of lists, but I feel like with Parks Canada, it might actually be possible to do something with this that's like like an actionable list for Parks Canada practitioners, because they are divided into discrete geographic areas, the parks themselves. So it seems to me that you might be able to compile a list, but then have like, okay, for like Banff National Park, here's a list of assays that we know for fish species present in it might be valid for this park. Here's a list of like that. That seems like a promising approach to me.
Yeah, that's true. And actually, I did that because we do have already some qPCR assays that have been developed, especially for Parks Canada issue research project that have been conducted in 2 parks. So yeah, so I did mention that in the column where I had the information where was developed the qPCR assays, and I did mention that in the list.
Yeah, and I'm so glad you're participating, because this is exactly the sort of feedback that we want, because I mean, this is what GEN-FISH is trying to do. It's just that we're lagging behind the need that I think that we have 156 assays developed now, something like that, but we're still in the process of documenting all of the things that we think are really needed before we can share that information. So we get, when we have assays developed for 156 species, but they aren't all validated yet, and so it would be disingenuous to say, yeah, you know, go ahead and use these. That at the very beginning of GEN-FISH, a student at the University of Guelph took all of the published eDNA assays for freshwater fishes of Canada and did in silico analysis to see which would be species specific, like universally species specific, and only 9% actually were truly species specific. And so there's always a caveat. When someone says, do you have an assay for lake sturgeon? We say, we have an assay that works for lake sturgeon in Manitoba when they're the only sturgeon, but it would cross-react with short-nosed sturgeon. And so that there's a caveat with most of them. Having said that, sometimes you can work around that at least you know, okay, you take it to another area and then it's going to detect one of two species. Maybe you need a second assay to then distinguish those two, or you need to refine the assay a little bit rather than starting from scratch again. So yeah, we definitely, yeah, because I think Paige had forwarded your email to me, and it was one of those things, we're working on it, and I wish we, you know, were further along to share it with you. But that's the sort of the ultimate goal, that at least, you know, most of the groundwork has been done by GEN-FISH, and then there's enough details included so that if we say we've got an assay for horny head chub, you know, then you can sort of take a look at the possible cross amplification and look at, oh, it potentially cross amplifies with two species. Neither of those two species occur in our area. We're good to go. So having that, having that additional information reported is what's critical. And one other thing I will add too is that a lot of our partners either want to use the assay without, you know, the months required to develop and validate, or even, as you had suggested, you know, collaborating with somebody else who will run the assays, and then you're acting on the results to make a management decision. I'm part of a group that Paige is part of as well, that's funded through the Great Lakes Fishery Commission Science Transfer Program, and we're putting together a checklist for managers so that they at least know the questions to ask their eDNA providers, because what has happened a lot before is that somebody will say, I've got an assay for northern snakehead. I'll run your samples to see if you've got invasive northern snakehead in your state. They run the assay, it lights up all over the place. They think they've got an invasion, and after the fact, they find out that it cross-reacted with smallmouth bass, and oops, sorry, sort of thing. But the managers don't know what questions to ask, and so to be able to say, you know, what did your in silico validation look like? Did you do in vitro validation? Did you sequence some of the amplicons that were amplified from the environmental samples to confirm that it was indeed snakehead? And this particular case, the managers definitely felt that they had sort of been burned by the promise of this great technology. But they themselves didn't know what questions to ask. And so now the idea is that we have this checklist that they can sit down with the eDNA service provider, or send them the checklist, and they can provide the information. And if they're saying, you know, no, we haven't done in vitro validation, no, we have, then those are red flags that should be raised.
Yeah, that's great actually to know, because that's exactly the checklist I'm trying to make for, yeah, actually in the document, in the step where they have to choose a collaborator or a service provider. So yeah.
Yeah, well, I think we're wrapping that up in the early spring. We had sort of a test run with some of the sea lamprey control people on Tuesday to sort of run through the pilot of our checklist. But as we're finalizing it, I'll make sure we keep you in the loop so that if the checklist itself is useful, or if it could be quickly adapted for your purposes rather than starting from scratch.
That would be awesome. Thank you.
Yeah.
Curious, in the Parks Canada document, are you featuring, like, or are you chatting to folks who've used eDNA? Because I've a number of my projects from my previous postdoc and current projects that I'm loosely affiliated with are working with the folks in Banff, like Shelley Humphreys and Brad Stitt, and I know they're going pretty hard on the eDNA for looking at like invasive species eradication. So it's nice to see Parks Canada taking up the tools kind of thing. So just wondering if you had chatted with them or anything like that.
I have not actually. You mean if I chat with everyone involved in that? Oh.
No, just with anyone involved in that. Like, are you looking to feature case
Oh, in
studies of
Banff? I don't think so. No.
Okay. Yeah. Right on.
But
okay.
there is actually
well,
lots. Yeah, sorry.
Sorry, no, go ahead.
No, I just wanted to say that there is a lot to cover in Parks Canada, because this is for the monitoring program, but we have also a program for the species at risk and for controlling and detecting the invasive species. So yeah, there is a lot.
Yeah, I can only imagine.
Yes. And I sometimes forget that there's more out there than just freshwater fishes too.
Yeah. Yeah. Yeah, that's
Yeah,
fair too.
it's funny that you are talking about eDNA with Parks Canada, because Jesse, who's also on the call here, and myself actually got to go up to Churchill this year to work with Parks Canada. There's Jesse, to do eDNA and teach them how to do eDNA. So
wow,
comes
wonderful. Where was it?
In Wapusk National Park.
Okay.
Yeah, so the Churchill, Manitoba, Parks Canada team, I think, and then we went into Wapusk National Park.
Awesome.
So speaking of application, I guess we're actually kind of nearing the end of our time. We're definitely in the second half. So I suppose, yeah, the kind of the second aspect of this is of applying these types of tools, not only designing and validating them, is applying them and actually making ecological inferences from these tools. So I'm just curious if we can continue discussing about design and validation, but I just wanted to throw out whether anybody in the group has any questions related to actually going out and applying these assays in natural ecosystems and potentially interpreting the data from it. As well, I also want to mention too, Brooklyn's presentation did focus on qPCR assay design and validation, but we're open to discussions of any tool, including metabarcoding and metagenomics, although GEN-FISH does a lot of qPCR and metabarcoding. We don't do any metagenomics as far as I know, but I'm sure we'd still be happy to chat about that if anybody wanted to bring it up. So yeah, if anybody has any questions or comments or anything related to that subject, feel free to chime in.
Yeah, because I think our main theme is, how do you develop these tools, and then what needs to be done to make sure you can use and trust them. So so those of you who are on the application side of things, we're definitely happy to hear from you as well.
Yeah, I guess I suppose I don't want to put you on the spot, but I'm curious, what's your perception of the sort of trust that managers in Parks Canada, like the folks on the ground, have in these types of tools. I mean, I'm sure there's a lot of enthusiasm for a new tool, but with that enthusiasm often comes kind of like a bit of skepticism as well for its application. So I'm curious what your perception is that you've had in writing this document.
Actually, I have to say that it's an excellent question, and I was not ready I don't know how to handle this question, but I was not aware about. So I've been doing genomic works for years and years. So for me, it was kind of easy to understand the capacity of eDNA for application in biodiversity conservation. But when I arrived in Perth, Canada, I just realized that it was not so obvious for everyone. And especially my own managers and managers related into the programs, they were kind of, okay, can you tell me how eDNA can be used in biodiversity conservation or biodiversity monitoring? And I say, oh, okay, you're already asking me the questions. So it means for me, it was kind of, okay, this part is done, and we all agree that we have to use this tool to complement the other traditional approaches. But this is not yet, we are not yet at this level. I mean, into Perth, Canada. I think other governmental institutions like DFO, they are in an extra layer, and they have, because they have got their own genetic lab and they are using it quite often, and they are monitoring it. So it's not, but Perth, Canada, because we do not have any genetic labs, people, they are not running themselves this kind of studies. So it's kind of difficult for them to understand all the tricky parts, all the details, all the laboratory works, and even the sampling sometimes. But you know, girls, that because you have been there. So, but for me, it was kind of, that's an excellent question, and thank you for that. But I was not prepared to that, and you see, few weeks ago, maybe 2-3 weeks ago, I had a meeting with a colleague, and it was the first time that we met each other, and she was saying, oh, I'm glad to meet you because my manager sent me your paper, and I just read it, and she said your eDNA paper. And I say, what? My eDNA paper? I've never wrote such paper. Yes, your paper you just published. And I say, oh no, oh okay, that's a big review in which we are talking about eDNA. But I wrote every other sections, but not the eDNA. So, and for them there is no distinction between eDNA and other genomic works and population genomics. So this is all confused, and I think. But we are getting there slowly, but we are getting there. So there are some people in the fields that have been already in contact with researchers doing that, and they see some concrete application into the parks for species at risk or for even the ecosystem approach, and they are convinced that we should use it. But there are some people that have never been confronted to this approach, and there is still some fear, because it's kind of new. It's not easy to understand sometimes. Even if I think eDNA, it's kind of easier to understand than other genomic approach. But so yeah, I'm struggling with that, and I think I'm not alone, but there is not many people into Perth, Canada that can talk genetics with and eDNA. So yeah, making this document is kind of a huge work actually to make it available for everyone and ready to use to everyone actually. Anyway. Yeah, I don't know if I answered.
Yeah. No, definitely. I guess that harkens back to Dr. Docker's point earlier about how there's a need to educate folks on even what questions to ask. Like if you're not familiar with genetics tools, you don't even have kind of a frame of reference with which to evaluate, can you even trust or distrust a result, because you don't have the knowledge or skills, I guess, to even evaluate that question without some sort of genetics background or training.
Yeah,
Yeah.
totally. And
Yeah.
that's why for me I have to make the opposite way. Because I have too much details in mind. I mean, genetically, like in the lab, and bioinformatically. So I can't see like easily their questions. I'm more used to, but at the beginning I was really struggling with that. So, but yeah.
Yeah, and it's tricky because I mean I've been working with eDNA for maybe since 2009, and so I think initially, and some people still do it, I think the tendency is to oversell eDNA, that it's faster, it's cheaper, it's this sort of silver bullet, it'll solve all your problems, without conveying some of the caveats that you have to be aware of. And then, because I mean that northern snakehead smallmouth bass example was a real example, and that there were some managers, I'm sure, that will never again use eDNA because there just wasn't the rigor in developing and testing the assay, or the transparency. And I know, like with there are some companies that are like especially for some of the stream side assays, that they have these proprietary eDNA assays that they don't even they won't tell you yet what the primer sequences are, because it's all proprietary, and it truly is a trust us scenario, and you can't make management decisions based on these unknown assays, and so there has to be that transparency. And I think what it's really going to take is, well, first of all, the more detailed testing and the reporting. But what I think what it's really going to take is the side-by-side comparison, because I know even with like seeing, okay, what does conventional sampling give us? What does eDNA sampling give us? It's never going to be exactly the same, but maybe you can see, okay, well, eDNA picks up the rare species better than conventional sampling, or the benthic ones, or the cryptic ones, and eDNA maybe is going to miss this or that, and so you have to be able to work as sort of partners on a situation that is of relevance to the end users, to see how well does it compare in our situation. A student at the University of Guelph that I co-supervised with Bob Hanner, Cam Brown, he's just finished his master's. He was using eDNA assays for sea lamprey, and then testing it with the sea lamprey control personnel on both sides of the border, sampling over 100 sites, I think, over 2 years, so that it's a big data set to be able to see how well does electrofishing and eDNA compare. Do they always give you the same results, but one's easier than the others, or are there some differences? And I honestly wasn't sure what we would find. I was pleased to see that it looks like eDNA is indeed more sensitive. It isn't necessarily easier or cheaper, but it does seem to be more sensitive, that in areas that are like above a barrier, that there was concern that maybe the barrier was a little bit leaky to sea lamprey passage, that it was picking up faint eDNA signals, or in areas that had been recently treated with a lampricide, no residual sea lamprey found with electrofishing, but eDNA was picking up a bit of a signal. So it's only with that side-by-side comparison, I think, that managers will be able to see the relative merits of different methods. Yeah, that the trust me just doesn't work very well.
Yeah, I will add, maybe if I can add something. For Past Canada, there is even an extra layer of difficulty. So there is this trust that people has to have with new technology, and also to have the trust of the partners, because many times there are some indigenous communities that are involved, for example, in the project. And I know that it was this winter when I just arrived in Perth, Canada, but I know that in Wood Buffalo, there was a project running. It was a GEN-FISH project running, but they simply give up to continue with this project because the indigenous community did not feel well included into this project, and they was feeling like the project was too science heavy. So they simply just decided to give up about continuing with this project. So there is an extra layer with people in Perth have to trust into the new technology, but has the partners has to do so. So yeah.
Yeah, and I found that too when I am working with various managers who haven't used eDNA before, whether it's parks or DFO. It's eDNA, I find, is like taking the lab into the field and how clean you have to keep everything, and how it's just not like typical field work. You know, when I'm doing electrofishing or gill netting with them, you're getting messy, you're getting dirty, fish are everywhere, all that kind of stuff. But with eDNA, you have to be so careful and so clean, and that's not something that they're used to as well. So it's very different than what a lot of them are used to. And I've found that doing it with them, it's helped a little bit in having them realize where there is that difference, and working through the steps, and explaining why we do things certain ways, and then how we have all these controls to see this is how we're making sure that our field gear isn't contaminated. This is why we collect this to make sure that this isn't contaminated. And this is why we change our gloves. This is why we use this bag. This is why we do this. That's helped a little bit, but it is a real big learning curve. And I know a lot of the time when we're teaching how to use eDNA and how to collect samples, they always seem to like make fun of me for yelling contamination or something like that, like, because I'm always like, no, that's contamination, watch out for contamination, contamination, because it's like I'm saying it constantly, but it really is such a different framework and way that you have to see things, and that can be so difficult when you're not used to lab work. So I can understand why there is such a difficult adaptation or adoption of eDNA, because it is so, it's not physical in the same way that you catch a fish, there it is, I know that it's here. It's all like sight unseen, as that classic Jared paper says.
Yeah, I can echo that too from my own experience. As I coordinate the Activity 2 survey, so I do a lot of trainings with like collaborating groups. Yeah, I mean, we've never had a situation where we've just handed a group a protocol and been like, here you go. I feel like that would be fairly irresponsible way to do eDNA, and also like have very poor uptake. But even differences between having in-person training versus something like a virtual training, like I have not had a ton of success. We're batting about 50% for having follow through with community organizations from doing virtual trainings. Like it works sometimes, and we'll have folks who buy in and do it. But even if you're doing virtual training with somebody about like how to go through the process of collecting the samples, and you're doing like a dry run, folks can get really, my perception anyways, is that folks just get really intimidated by the idea of collecting eDNA and the sort of like complexity with ensuring contamination and stuff. And the thing is that if it's in person, it's actually not that bad. When you can show somebody like how to do it in person and just take them to a side of a stream and be like, yeah, you know, take half an hour, collect some eDNA samples, we'll be good to go, kind of thing. It's much less intimidating, but yeah, that in-person training makes a huge difference. And like actually just walking through practically the steps it takes to take like a real eDNA sample is so much more effective than talking about it in kind of an abstract, theoretical classroom way.
Yeah, and building the relationship and saying like, oh yeah, the stuff that you see on the filter, then we take that the eDNA's there, we take that, and then we can tell from that whether they're there or not. And I find that that's been a lot better than this like explanation where it's just all like voodoo magic where I take water and now I know if a fish is there. So it's been much better.
Well, we've got about 2 minutes left before we got to go back to the main room. So I guess we can open the floor. Joe, you got your hand up.
That was why the hand was up. So looks like the gang from the STP room have migrated over. So I will give you the 2 minutes, but then we'll shut this down so we can get the recording, and then we'll move over back to the day one. Thanks.
Okay. Sounds good. Well, thanks everybody for participating. I guess if there's any last questions, if anybody has them, speak up or forever hold your peace kind of thing. But otherwise, we probably should head back, I guess, now that the STP chip discussion is concluded as well. And thank you, Anne, for participating as well.
Yeah.
I think you brought a really interesting perspective to the discussion. Your paper on effective and census population sizes is really good, for the record. Fantastic study. Anyways, all right. I'll see you, everybody else in the main room.
Okay. Thanks, everyone.
Yeah.
Thank you very much.
So I'm actually going to Turks and Caicos next week. I'm going to suss it out for our next AGM. We'll go to Turks and Caicos.
Remember that short-lived period where there was some thought that they might become a Canadian territory? They were looking for a larger friend.
Yep.
Fortunately, somebody fumbled the ball on that one. So
yes, our federal politicians got all excited about things about, I don't know, having to provide social benefits to all those Turks and Caicos inhabitants. Missing the point entirely. Okay, shall we start?
Yes, all the breakout rooms have been closed.
Yeah. Good. Tina, do you want to start us up?
Yes, of course. The reason why we're coming together is to collate all of the issues that have come up in terms of what we'd like to see going forward, whether it's in the next 18 months, which is when GEN-FISH will close the doors and close the book, or in terms of what our end users are looking for. So how are we going to get to the end of GEN-FISH, but also what are the next steps? Because again, the whole point of the deliverable of GEN-FISH really rests in activity 5, and that's that decision-making toolkit. That decision-making toolkit is really encapsulates everything that we're here and we're discussing today. It's all about what can facilitate the adoption of whether or not it's the fish survey toolkit, which is the eDNA, or the fish health toolkit, which is our STP chip, having our end users in mind and hit the questions of biodiversity, conservation, and mandates, whether or not they're coming from government, provincial, or federal, whether or not it's from our industrial partners, whether or not it's from our ENGOs, whether or not it's from our community practitioners. So if we can get a summary potential, and this is going to be really helpful for our Activity 5 members as well. So, it's not just self-serving for Activities 1 through 4, but it's for Activity 5 as well. And I was wondering if we could start potentially with just a summary from Activities 1 and 2, and then Activity 3 and 4, listen to see were there any commonalities or is there any distinction? And then we could also have a discussion on how we would overcome them. So Margaret or Matthew or whoever was co-leading that, please go ahead.
Yeah.
Sorry, I apologize. I had to step out to take a phone call, but we're just summarizing the previous session, correct?
That's right, and identifying any solutions you came up with or any types of barriers as well.
Well, I think honestly, the one of the major barriers that sort of kept coming up in the last session was kind of the need for education, both in terms of like the session we had, we divided up into two sort of sections, a bit of an arbitrary distinction, but the first half was focused on assay design and validation. The second half, we kind of talked about applying them and actually like using eDNA assays in practice. And a key thing that came up is that a lot of the times folks and managers just don't even like we were talking about trust in the tools and sort of the applicability of the tools, but a lot of the times managers, I guess eDNA is so new that a lot of the times managers and the folks on the ground who are using it oftentimes don't even have the frame of reference or baseline knowledge to evaluate how appropriate a tool is, how best to apply it, or even to, like, you know, properly interpret or trust data that are generated from it. So I think that was one thing that kind of emerged out of that discussion, was that there's kind of a genuine need for improved education. And Dr. Docker mentioned a kind of a tool, part of a decision. Well, actually, Dr. Docker, you probably would be best to discuss this one as well. But basically the guidance document that you guys were designing for folks to, you know, what kind of questions even to ask in terms of implementing eDNA assays and using them for managers.
Yeah, and I think that we spend a lot of time talking about how, you know, I think that in the past, people have sort of oversold the eDNA, and it's quick, it's easy, it's magic sort of thing, and without providing the information that the managers need to be able to trust it. And so we talked a lot about the importance of transparency and reporting. I mean, the really rigorous testing. So all the things that GEN-FISH is doing, not quite ready to share with the public yet, but we're doing all of that. But then in terms of that toolkit that we're developing through the Great Lakes Fishery Commission Science Transfer Program, that Paige has been working her magic on to develop this really, really nice interactive tool that managers themselves, I mean, that we can definitely ourselves do a better job at explaining, you know, what is involved with the assay, what the caveats are in terms of, okay, well, it's, you know, it might still cross-react with this other species. I mean, those sorts of caveats, that we can definitely do a better job at that. But at the same time, the managers themselves aren't going to be the experts in eDNA. Sometimes what they just need to do is know what questions to ask the eDNA service provider. So if they're sending it to the samples to a consulting company or something, they at least want to have a checklist of what they should ask. And so that's part of this interactive decision toolkit that we're putting together, where they can ask, you know, how many technical replicates, you know, has species specificity been tested? Are amplicons being sequenced to verify species specificity? That there's at least that list of questions that they can ask and get information. And it's really nice the way Paige has set it up, where if the answer is, you know, no to a number of those questions, you know, there's a red flag that's raised. So it means that managers know what to ask, have a good sense of how reliable the results are without they themselves needing to be experts on it.
Now, do you think that the managers need to ask in terms of what has to be done for sampling, or in terms of trusting the validity of the results? So there's the prior to the sample checklist potentially, and then there's the post. It's the data, you know, validation and verification analysis. So where do you see the checklist coming, or both sets of checklists?
I'd say both in all of those things. That I think it's set up in different sections. I think there's the first is, you know, the pre-flight checklist with talking about sample design and, you know, what's your goal? Is it sample presence or species presence or species absence? I mean, just basically to address things before you even start working out the details, and then field collection and lab work, and it's also sort of modular, so that if you're doing the field work yourself, you can work through that checklist, and if you're sending your samples off to a lab, they can fill out the lab part of the checklist and send back the answers. But yeah, the idea is from start to finish that QA/QC is done all the way through in a fairly streamlined way. I mean, as we know, like some of the standard operating procedures for eDNA could be like 80, 100 pages with all the details. So it's has to be a balance between enough information to be useful, but not so much information that it's overwhelming.
Yeah. All right, thanks, Margaret. I know, Chris, you had your hand up, but before that, I just want to reiterate that Amy said that a lot of the issue of trust or mistrust has come up in Amy's focus groups as well, and that was shared across multiple stakeholder groups. And the issue of trust was also brought up today in Steve's co-production workshop as well. And so by able to co-produce, there's that trust that's being established in terms of the knowledge that's being exchanged. And so I think that's a really, really interesting thread. And what I'm hearing is that information provision and discussions and workshops ahead of time can contribute towards that and having a streamlined process. Excellent. Chris, did you want to add anything? Because I know you had some comments in the chat as well. If you want to say them out loud too, please go ahead.
Okay, sure. Just briefly, Margaret's given a great overview of the GLFC tool that's being developed. And the thing is, because it is modular, not all users need to access all parts of it. So, but criterion one is there a clearly defined question? Number two is, does the sampling design and effort reflect the question? It's like Margaret's mentioned sort of presence versus absence. In the fisheries world, that's one concatenated word. It's always, oh, presence, absence. It's like saying black and white are the same thing. They're two diametrically opposed questions. So depending on the question, sampling design and effort really needs to reflect that. So this is little things trying to educate end users. But also I just wanted to mention that you're probably aware different agencies across the country are at different stages of trying to grapple with eDNA, sometimes within the same agencies. So for Ontario, Natural Resources is using it. It's basically the first early warning system for invasives. It's being used on an ad hoc level for endangered species because endangered species are now with the MECP, or Ministry of Environment, Conservation and Parks. MECP now has a position paper on eDNA that Scott Reed with the M&R, silent F, and I helped them construct. So there's varying acceptance within agencies as well as across agencies. There's a lot of interest, but I would say overall there's still low confidence. And DFO has done a wonderful job of trying to come up to speed. So with their national working group, and they have some very good guidance documents that are being referred to as resource materials for this GLFC tool. And also those documents were very influential in terms of developing the CSA national standards for eDNA. And as far as I know, sorry, Matthew.
Sorry. Yeah, my perception of DFO is that they really are almost essentially an internationally leading government agency in terms of the adoption of eDNA and its practical application. Like they are really well advanced, far advanced in terms of working through these problems as an agency.
They are doing very well. I would actually say Australia and New Zealand are ahead of everybody in Canada put together.
Fair enough.
And the US is going to blast right past us within the next 12 to 18 months. But that's good because then we can follow. So that's a little bit of background on agency perspective. They're all grappling with it. DFO is doing a very good job. M&R is mostly there. There's still the show me the fish and I'll believe you. But in the meantime, they want the results and the interpretation. And as somebody inside, I'm very pleased with how they're using this for invasives, to the point where people are going out and searching, not finding the species of interest, but the eDNA says keep looking. They're actually keeping looking, which is wonderful. So, and I will avoid tangents. Anyway, that was it for me.
Oh, thanks a lot, Chris. Any other comment or any other discussion topic points that you hit on in your workshop?
Maybe just I'll quickly follow up on that, because then I know there's other things to talk about too. Just going back to that mistrust, and mistrust was brought up a lot in activity 5, and I think again, like I think early sort of eDNA adopters, like did the field a disservice by overselling it initially, and where we see eDNA really taking off, it seems, is like with DFO, with agencies that are doing all of their own work. I think the mistrust is still with the end users who are relying on others and are being told that, you know, well trust me, you know, it works, without being able to see it side by side. And I think, I mean, I'm hoping that we're doing the right things in GEN-FISH, where we're really trying to develop and validate those assays, reporting all of the information so that people can use their discretion as to whether it will suit them. But then in activity 2, with those side-by-side comparisons time and time again with paired sampling, and so that end users can see, okay, now I see that it works in my lake or in my river or in my park. I don't have to trust you that it works, and I can now see where it works, where perhaps it doesn't work, and then there's that ability to make decisions better.
Mm-hmm. So how do we then translate the findings of activity 2 in a more global space? So with our practitioners, I know we're going to have standalone papers. We're all going to have standalone papers, but is there some way that, and we had talk in activity 3 and 4 about collating the data itself, so that we could analyze and look for patterns. Is there something that was discussed in activities 1 and 2? So 2 things: collating all the data so that we could show general patterns that, yes, this is how it's validated. And then how do we get that information out to our end users? So Margaret and Matthew, you're not on the spot for that, but it could be a more general discussion. And then we can tie that in also with activity 3 and 4.
Well, maybe
so
I'll just,
repository.
yeah,
I'm just trying
to. Yeah, I'm just trying to think, like, Matthew, when we were talking on Tuesday, I think you were talking about like a Venn diagram of, you know, species that are detected with conventional means, species that are detected with eDNA, you know. Hopefully there's a lot of overlap, but understanding where there isn't overlap, and so that, and you've already sort of alluded to that synthesis of all of the different case studies in activity 2. So it's not just individual case studies, but I'm hoping that by the end of this, that we can get a better sense of when will eDNA outperform conventional, under what circumstances, when will it give you the same results, when should you not rely as heavily on eDNA. That doesn't, I don't think it fully answers your question, but maybe gets part of the way to answering your question.
Yeah. Chris mentioned your STP tool with the through the GLFC, and I know that we're doing that also for activity 3 and 4. So Margaret, can you just give people on our workshop right now just a bit of a background and what Chris meant with your STP tool, decision-making tool?
Yeah, it's a checklist of all of the things that you need to consider when you're using eDNA, broken down into those different modules. You know, the pre-flight checklist, where you're looking at the sample design and the goals, field work, you know, assay design, lab work, as a way of managers who want to use the eDNA results, that they know what questions to ask to evaluate the robustness of the methods.
So I see there's prongs of information. And just to let you know, STP in this case doesn't stand for stress transcriptional profile.
Yes.
It's a strategic trans.
It's science transfer program.
Okay.
So,
Thank you.
yes, and I've just noticed that Paige has put a link to the static checklist version of the tool. So she's been working on this, and that it's meant to be interactive, that you can work through it and check things off. And there's little information boxes that pop up. But she's also shared the static version of it. I don't know if it's easy for us to look at that on a shared screen or not.
So I see 3 prongs of information coming out, and again, it can be universal. The first one is your pre-flight checklist prior to sampling. I know it's going to be tailored for different agencies that require different amounts of information. Then there's the post-analysis of all the data in terms of, okay, should I use this, yes or no? What should I check in terms of the sequence, the QC? And is this valid, yes or no? There has to be that kind of education information. And then I think the third one is, like you said, this kind of meta-analysis on all the information coming out. So yes, we could look at the output of our eDNA analysis, and we could say, okay, like Hugh was talking about, false positives, false negatives. But beyond that, it's okay, how much trust should I put in the data, regardless if it's telling me it's past a standard? And that almost comes to more of a social issue, social ecological issue as well. Those are 3 really important key pieces of information that I think can be incorporated into the transferability and the adoptability of the tool. And I don't see this actually being that distinct from the STP-Chip. So the health, I got Steve putting up his hand, our health toolkit. I think though, we're, how Chris said that Canada's still behind New Zealand and Australia. I think when we come to the health tool chip, I think we're dead last, but I think it's just as a concept too. But I'll pause there, and I'm going to switch it over. Yeah, Steve, what are your comments?
Yeah, I'm interested to hear from Chris Wilson again on the extent to which there's discussions about, before you sample, what if? What if you get a positive? In other words, working through the management or response scenarios ahead of time to know how the response is going to be, instead of waiting until you're surprised and then the go-to is, almost like a defensive approach, right? So yeah, anyways, interested in that, where things are at.
So Margaret and I were involved in an earlier eDNA science transfer project, years ago now, that's mirrored on the GEN-FISH site, where it talks about uses and limitations of eDNA. And one of those was basically a flowchart as a decision support tool. So saying, depending on the results, how do you want to put this in context? What other actions might you want to consider? So that's already out there. This one is more, how much confidence can you have in both the study itself, the data generation in it, and the interpretation? So the first one was saying, okay, you've got a positive result. Let's put it in the broader context. This one is saying, can you trust the results? So can you infer species absence based on collecting one sample from Lake Erie? Probably not. No, species presence, maybe. Species absence, non-detection is nowhere near a true zero. So there's why I keep leading with that front end of, does the sampling effort and design reflect the question? So this would allow an end user, somebody with no background in this, saying, have the right questions been asked and the thresholds passed for each step of checking reliability, to say, okay, this is reliable information. Now I can decide how I want to use it. But it's basically, can we establish that trust? Margaret, did I capture that? Or Paige?
I would say so, yes.
Margaret, did you want to add to that?
Yeah, and I really like the way Steve phrased that, because I hadn't really thought of it that explicitly before, about, basically thinking in advance of using eDNA. What would you do if you get a positive hit for eDNA for, say, an invasive species? If your response would be, well, I'm not going to believe it until I see the fish, then that's something different, maybe eDNA isn't what you want to do, or if it's going to be, okay, well, the eDNA is going to help us narrow our search as we look for confirmation with a physical specimen, and getting a sense of what the goal is and what your response would be. And then that ties in, with the first science transfer program project, where emphasizing a couple of things. That first of all, that a single detection doesn't necessarily bring a management response. Like if Brooklyn was out in the Red River and detected sea lamprey, would we apply TFM to the entire river based on one sea lamprey detection? Probably not. That it would then trigger additional follow-up sampling, and then also having to balance, depending on what the question is, what's the cost of a false negative versus the cost of a false positive, that how much can you afford to miss the species being detected from a management point of view. So having those discussions all sort of in advance of embarking on the project are really important.
So I'm going to now kind of shift to activity 3 and 4 and ask what were some of the barriers or solutions that were discussed, but also keep in mind what we just ended on in terms of trust, going ahead, looking at the design of the project, what's people's mandates, all from the point of view of fish health. So Daniel or Ken?
So Ken, I'll start. I think the STP-Chip projects are perhaps a little further behind in terms of worrying about barriers to adoption, simply because the technology is not something anybody else is doing. So eDNA, there are a bunch of agencies that are working on it. We were really working at the ground zero to develop it. And so a lot of what we discussed had to do with technical hurdles before even we as scientists would really adopt it, much less get end users. One thing though that just, listening to Chris, Margaret, and Matthew, is there's I think a fairly fundamental reluctance to accept eDNA. As I put in the chat, most fish people are very comfortable with holding a fish in their hand. On the STP-Chip side, it's really hard to tell if a fish is sick until it's dead. And so there is actually a lot of interest. I am getting partners contacting me about wanting to run chips, even though our chips aren't validated, simply because they want an objective measure of whether a fish is stressed or a level of well-being. And this got tied into something that you brought up, Tina, that I thought was a really, really nice way of putting it, is whether or not the chips will be prognostic or diagnostic in terms of their applications. And our intent was for it to be prognostic. You look at a fish that looks perfectly healthy and you find out that it is struggling. If we can show that with a bunch of controlled studies, I think adoption will probably be a lot less challenging than the eDNA. Having said that, the technology is terrifying. To many of us who do transcriptomics, the technology is opaque. So I think we need to come up with interpretation tools, and they need to be user-friendly. They can't be enormous R script with, very difficult user interfaces. Ken?
Yeah, that was great summary, Daniel. I think one of the things that we talked about, again, it was much more from a technical aspect, and how are we, making ourselves more comfortable with the data produced. And there's really good discussion about how we can compare the gene chip results with other more commonly used methods for looking at gene transcription. So things like comparing the gene chip response with RNA-seq, for example, or looking at things like protein level responses and whether that matches well with what the open array chip is telling us. And that way we'd have a preemptively a better method of trying to show that the chip data is representative of a response in the fish as well.
And my question is, how does that then translate into trust in the technique itself? So we show we validate it, and potentially to a practitioner end user, they're just going to have to take our word for it that we used RNA-seq and it validates. And so where do you think that trust level rests in terms of we've used multiple ways to show that our chip is universally saying the same thing. So I think and I'd love to hear people's opinions on that. And I might just say, we also have discussions, and Brian Dixon on the call had said, you know what, we can show potentially going from the mRNA all the way to protein synthesis. But now we're doing proteomics potentially, but showing that it can carry through to have a system level response or individual level response. So we also need to have that level of confidence, and with uncertainty, obviously, that if we show something prognostically, what's the likelihood of it actually becoming an issue? I think that there's a lot of uncertainty in there, and it's going to take a lot of convincing, or maybe not. We just leave it in the user's hands and say, this is what we're saying, it's up to you. I don't think it'll have that much of an adoption. So there's that issue too. I had Celine with her hand up first. Oh my gosh, everything's lighting up. Celine, I'd love to hear from you first. And yes, please go ahead.
Well, I just want to say that it comes back to a lot of things that Steve presented earlier this afternoon. I just want to talk about the experience we had with the strip bass data that we try to use the PERSID chip. And we work with MFFP, the provincial government here on FONA, and they were not discouraged at all. They just called me and know if GEN-FISH would be interested to test chips on other species. Then, as long as we work with our partners and that we are really, I think that it just come back. I mean, the trust is in that. I mean, the way we work with them, that we are transparent with our data, and my experience is that they were not afraid at all. As they are supposed to reach Danielle and see if there could be some interest to work with other species there. I'm not afraid about that.
That's amazing. And that's a really good message, Celine. Thank you. We already have the reason why we've all been chosen in this network is because of our connections and networks to end users or users in the field. And we have these working relationships. And so hopefully we're going out there and establishing trust either through our students, ourselves, our connections, and we're facilitating that with our future HQP as well. Let's keep that in mind, again, because it comes back down to a social issue. I'm going to stop because I want to hear Mark and his thoughts.
I think it's very interesting. I think we're dealing with almost two different groups of end users. And I think back to when I was first starting my PhD, and Craig Clark developed the 24-hour saltwater challenge. And before that, I used to take the smolts and just dump them into seawater and see if they died. And then he said, well, we can take plasma from them and measure plasma sodium. And there was a fairly quick adoption of, hey, we can just get a number out of a machine that'll give us some indication. Whereas my experience talking to people in industry and government agencies is, I like to see the fish in my hand before I believe that it's actually in the water. And comments like, oh, well, eDNA doesn't work. There's, 30 to 40%, error rate, numbers that I just don't know where they come from. So I think there's going to be a much easier acceptance of using technology to address physiological or stress or transcriptomics type questions than there is this fish here or not? So I think we have quite different audiences.
Yeah, that's a really, really good point, Mark. Chris, what do you have to say?
Actually, I rearranged myself so Steve could go first. I think he's more squeezed for time.
Steve, sorry about that. No, that was my bad. Go for it.
I'm good. I'm home now, so I'm good.
Oh,
go ahead.
Okay. Okay, then I'll stick myself back in. Take my hand down. Just I work a fair bit with the fish culture group within the MNRF. There is a lot of appetite for this, for fish health screening, and a high level of trust already. They're used to sending obviously sick fish or mortalities off for fish health testing. And so they've been able to keep up as the technology is bridged from histology to PCR based. So they've had that learning curve. They want this information. There's a very strong appetite for this, for both prognostic and diagnostic, because they have a lot of at stake for this. So if there's basically an early warning, that gives them the option of prophylactic treatment to prevent the problem. And there's routinely diseases will crop up from time to time. I actually think there would be appetite to help test this. They spend close to a couple of $100,000 a year just for fish health testing. Basically, incorporating this would give them huge information return at a marginal cost increase. And likewise for the aquaculture industry, Brian could speak to this better, but for them, their livelihood is at stake for this. So again, this is really valuable information. So as Mark said, I don't think there's the same trust issue, and instead there is a very large appetite for both industry and agencies for this. So I think the trust is already established.
So, and I'm Steve and Brian, I want you also to contemplate this. Do you think that then, and Chris, there are easier sells with that prognostic tool? Because again, immunology can speak. You said disease. Ken loves pollution. Those are pretty strong markers of, yes, these fish are in trouble. But we've also chosen other genes. And Nick Bernier, maybe you can speak to this a bit too. Because we looked at when we were doing the grant, we looked at, okay, what are the major stressors that are affecting Canadian freshwater fishes? And some of them are invasive species. So that's why we went after stress biomarkers. And we know that whether or not stress goes up, stress goes down, does it mean that they're compromised, or are they actually mounting an increased stress response? We actually have appetite genes to show whether or not they're in increased competition potentially with invasive species. So we also have to think of the layers of the prognostic indicator.
Oh.
She froze, yeah.
To keep in mind. Hard. Okay, Steve, what are your thoughts about that? And other thoughts, go for it.
Cool. So I just posted a few papers in the chat there that are some of them in that space of linking what happens at the level of the individual or sub biological level to the population. And this is, I think, sort of where the rubber hits the road with all of this stuff. Resource managers manage populations. They don't manage individuals unless the population is so pathetically low that you're truly managing individuals. All of the examples that were just given, which were lovely examples, are all in that fish health space by people that are already used to working with individuals to understand population level processes. And maybe that's because of an epidemiological way of thinking or whatever, that's normalized. The routine, and Chris can tell me if I'm misreading how my M&R colleagues operate, but the average district biologist / resource manager doesn't care about sublethal or, so unless there's an issue, unless this lake, there's something weird going on, we need to sort of sort it out. So I do think thinking about how to scale, especially omics, as soon as you start dealing with omics, it's even more steps removed from the individual. Again, it's almost like identifying case studies. And I know you've tried to do that, Christina, in your previous paper, but we're still missing the routine fisheries manager that cares about abundance and managing people, habitat, fish to make anglers happy, to ensure that we don't overfish populations, to, that side of things, figuring out how to insert ourselves into that community. That opens huge doors. That's the challenge.
Noted. That's a tough one. Brian, what are your thoughts?
I was going to actually make the point that Dan made in the chat, but in the fish health space, I think, as everyone said, there's a lot of uptake for this. I'm working with Center for Aquaculture Technologies and PI, and they want me to give them an ELISA to measure specific responses to specific pathogens because the companies are asking them for it. They want to know if their Forte 4 vaccine is working. And if it's working to all the components of the vaccine, I think that's going to go very badly for Elanco because there's going to be like massive response to one pathogen and nothing to the other. But they're very interested in the technology, and that company then markets the service to other aquaculture companies, like the producers that are asking it. So I think there's a level of trust there, and I think if we get companies like CatC to adopt it and offer it as an analytical service, and I think there'd be market for the chips as well if we can validate it. I think that would be very good. The danger is, as Dan said, we don't want to come in and say, here's the silver bullet, measure with this. It'll tell you everything about, like, I think the critical thing is back to what Steve said. We have to have the level of trust and treat it as we're beginning an experiment with these companies, and they're involved in the validation as well, right? To make sure that we're all on board with it. So it will take trust and time, I think, but I think the willingness is there, at least on the fish health side. So, yeah.
Good. And Chris, you've been giving some really great comments in our chat, but I won't read them out. I'd rather hear you speak. So go for it.
It's in the chat.
Your hand is up. You could contribute something new. Go for it.
No, shit. No, I'd just be repeating myself, honestly. The from the agency perspective, yeah, the sublethal, as long as the population is there, the local biologists on the ground and managers are just going to say, you know, we've got larger issues to deal with. They've moved away from managing individual populations to managing them collectively as sort of management zones. But there is the recognition that we do have increasing and compounded stressors. So stocking is going to become a more and more important tool, which is really unfortunate in my view. We have been the envy of, well, Ontario and Quebec have been the envy of the rest of the continent. As we're seeing increased stressors from overexploitation, overdevelopment, so land use changes, and climate change related stressors. We can expect to see increased stress within populations, increasing recruitment failure, and make them more and more vulnerable. So that's where the prognostic information would be able to say, these populations are increasingly under stress. Can we in advance modify the regulations so we don't lose them? Stop them dropping below that threshold or tipping point where we now have to maintain them artificially. That will be education. There's been an ongoing effort within both DFO and the MNR to educate the decision makers for, I'd say, the past 5 or 6 years. And actually, Trevor has been a big part of this. He's on the most trusted status list for MNR's fish culture group.
Cool.
And oh, sorry, Trevor, go ahead.
No, I was going to say, just to add on to Chris's point, and I was just in transit, so I couldn't do my camera, but fish culture, I just talked to them again. I don't do the health part, but I talked to them about these issues. They're very much interested in these technologies, and between Chris, myself, and a few others, I think we've identified this. We have some trust, but they also want guidance. And those guidance issues we talked about half an hour ago is going to be critical before they ever adopt anything. So I just see this being like a holistic problem that we kind of circle around with trust and then back to the guidance and back to the result not being overpromised. And it seems like a very clear dynamic that can be outlined fairly clearly for them.
And I definitely see parallels between our two fish health toolkits and in terms of the guidelines, the trust, pre-information, post. So before I switch to you, Daniel, just to say that, yes, we also have our science transfer program for the STP-Chip that Paige is also involved in. It's still in its nascent stages, but it's targeted, I believe, to fisheries managers, and Paige, and please correct me if I'm wrong, but it's all about that adoption and the needs. and the information that's required. And I do hear that there's uptake, but then again, there's uptake for the tool, but whether or not it's going to translate into management action, that like with a precautionary approach, that's a different story, and that's not up for us to solve. But because we can't oversell too much, but we still need to sell something. So we need to sell that we can say something about the health of the fish. It's at the individual level. They're going to want to know, again, what's our sampling regime? How many fish do we have to sample to say that this is a population-wide area? How is it linked then to identifying habitat structures, or turn it on its side, recovery measures as well? So we're going to have to try to think about what are the kind of thresholds we're going to be putting on the data that we're offering. Daniel, did you want to comment on that or anything else that's been happening in the chat and through conversations?
Well, this will be quick. Steve brought up a point that I had not thought of, and that is the application of this to wild populations is going to be the challenge. We have the advantage that fish culturalists will be comfortable and will provide sort of a proof of concept with known outcomes, which will translate, I think, well to wild fish applications. But I do think that it's the wild fish applications. One key point that I made earlier is the difference between RNA-seq data and the STP-Chips. And I really believe that these nanofluidic chips are going to make the transcriptomic data accessible for fisheries applications in a way that RNA-seq or proteomics or metabolomics or all the other omics will never get there, because there's just an opacity of that kind of data that will make people uncomfortable, make me uncomfortable, and I deal with it all the time.
Thanks, Daniel. Nicolas, what do you think?
Well, once again, going back to the application for wild fish populations, if we're thinking of this as a fish health toolkit, the STP-Chips, that is, one thing that I don't think we've entertained too much or discussed is the idea that this can only be useful for wild fish if we think about using it without control fish, without a control population, where you just go in the lake, take some fish out, and you want to know what's the health of these fish. And this is a real departure from the way we think about these things. And so I challenge all of us to think about this data as interpreting, going beyond our current interpretation, but looking at it in terms of ratios, in terms of, if you have 52 genes, is there a profile or there are ratios that are more important, that are indicative within a population? If you have, for example, immune genes elevated versus other genes within a fish, can you extrapolate from these 52 genes without control data and be able to say anything about the health of the fish? That's a real challenge, and something that we need to wrap our brain around in the next year and a half.
Daniel, do you have a response to that? A ray of hope to offer?
Well, I noticed that just about every time I do a Teams meeting with Nicolas Bernier, I disagree with him. I think if you think of the human situation, there's never a control, and yet they use this type of data to determine whether you're healthy or not, because this is the average baseline. Now, it's being highly criticized because the baseline are white males, but it's an approach that has worked. And I'm thinking that if we get to the point where we start looking at patterns of response, not interleukin-8 is downregulated, it's a sick fish, but maybe, as you say, a pattern of upregulation of immune function genes and a downregulation of metabolic genes, this is characteristic of what we see when we stress the fish. Because if we can't do that, then our fish health toolkit will not be applicable to wild fish where we don't have controls.
And yeah, I think we both agree. We're just not doing it. That's my point.
So we've got homework assigned to all of us then. And so we've got a minute to wrap up because I think it goes till 4:30. But I just want to impress upon everyone that yes, we're doing our own little projects, but we have to come together and we have to, at the end of the day, create these toolkits that will be accepted. And so we're going to be having greater conversations. I think activity 3 and 4 already decided to get together on a monthly basis just to talk about some of the issues and that are arising. We're still behind where eDNA, like activities 1 and 2 are, because we're still stuck in the technical weeds. But I think this shows that we've got discussion points going forward. But we need to start having these types of discussions to know how do we translate our tools. Again, the pre-flight checklist, the post-analysis, the trust, the information, and the usage. So all of that's really, really important for us to keep discussing, because at the end of the day, this helps the ultimate toolkit of GEN-FISH, which is this decision-making toolkit. We saw Peter give an excellent presentation this morning. There's so many more variables that can feed into that. He talked about efficiency, he talked about cost, he talked about time, but that's not really knowing what you know and what the end users know. And so we will be, and again, information for Amy Fitzgerald, information for Tongji, information for John Livermont. We're going to all have to come together and work on this collectively as well. So be prepared to have more meetings and workshops, because this is the mandate of GEN-FISH. Regardless of Genome Ontario and Canada oversight, this is what the purpose of the grant is. It's about the adoption and the transferability of all of our hard work. So stay tuned. We're going to keep discussing it. We're going to even talk about it more tomorrow in terms of where we are in our progress, so you can see it all in a gestalt way, in a collective way. And we're going to keep these conversations going because they're absolutely critical. Any last thoughts? Okay. Oh, okay then. So as we're all stunned into panic, thank you so much, everybody, for this wonderful day. Thank you to our HQP who are still on the call. I really enjoyed listening to all the talks and all the progress, just like Paige had said, that you're doing on behalf of GEN-FISH and helping out, again, our natural freshwater resources. And I look forward to seeing you all again tomorrow, bright-eyed and bushy-tailed at 11 EST. Check your, yeah, Steve, was that? Oh, I'm like, it's 10 o'clock, Tina. It's 10 o'clock. It's not, it's 11 EST. And until then, à bientôt, à demain. Until tomorrow, bon appétit. Thank you, everybody. Take good care.
Ciao.
Bye, everybody.
I should have raised a glass, Tina. You would have recognized that. So, yeah. Thank you.
Bye.
Bye.
Bye, everyone.
Thanks, everyone. Great first day.